Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I'm back with the Re-NEET Bounce Back series, and today is going to be a very important session guys, because in today's session we are going to discuss the most important and dreadful topic, that is genetics, right? So this session is totally dedicated to genetics 1 and genetics 2. That is, we are going to discuss two topics of principles of inheritance and variation and molecular basis. So heads up, and quickly like, subscribe and share the session. And for next 2.5 hours or maybe 3 hours, we are not getting up, we are not taking any break. But before we start with the session guys, please take notice of this, that all India mock test series has already started, happening thrice a week, and the next test is coming up on Sunday, that is 7th June, right? So next test is on 7th June 2 p.m. to 5 p.m. Please enroll for the test, because this test will give you exact analysis of your performance. So it is exact PYQ replica, NEET experience you will get, detailed performance insight where you can analyze your performance, because there will be video solutions also, and you can of course improve your rank by as much practice as you can. That is the only way out. Good evening all of you. Good evening guys. All right. Biotech is easy. Biotech, you will find it easy after you are very thorough with all the formulas of fundas of genetics. Your flow charts enough to score 340? Yes, Jyoti, they are enough. Or should you read NCERT along with it? I am trying to cover NCERT as much as possible. And second thing, after completing our marathons, I am going to take 2 long sessions where we will be reading NCERT. We'll be highlighting the important lines of NCERT. I did that before 3rd May also. I'm going to do that now also before the Re-NEET. So don't worry about that. And shall we get started? Please give me a thumbs up that you're ready, and we can start with the session. Yes, guys. Right? All right, guys. All right, cool. Now let's start with genetics. First of all, what is genetics? So we know it is a branch of biology that deals with two things. Biology that deals with two things. Number one, inheritance, and number two is variations. So what do these two words mean? Inheritance means transmission of characters from one generation to another. Why do we look like our parents? Why do we inherit their why do we inherit their characters, right? So it is inheritance of characters from one generation to another. And second, how and why are we different? So variations are the differences. Variations are the differences. Are these clear to all? Right? Okay. Now we are going to talk about these points one by one. Let's start with the details. So who led us into genetics? It was basically a mathematician. Yes, it was this Austrian monk who loved maths. So he was a mathematician. And now this one thing will become probably an obstacle in his way of success, right? We'll see how. So he's an Austrian monk, mathematician. He loved gardening, and he had grown garden pea in his kitchen garden. So he had a big area in the monastery, so where he was growing garden pea, also known as Pisum sativum. So this plant became his experimental organism. So this was the experimental plant. Are we clear? So garden pea or Pisum sativum. Is that clear? Now, what are the type of experiments that he performed? So he performed basically, performed artificial hybridization experiments. So these are the type of experiments that he performed. All these are one-word questions. right guys? So this was Gregor Johann Mendel. Now let's see. So remember now, this Mendel, he performed this experiment. So these 7 years, he gave 7 years to this experiment, 7 years. So remember, 7 is going to be his lucky number. He gave 7 years to this experiment. Also, he studied 7 characters, and out of these 7 characters, guys, 7 traits were dominant and 7 traits were recessive. So he studied 7 characters of pea plant. See here. Now this is from your NCERT itself. So see, what were the characters that he studied? The characters that he studied were, so this is the character. So characters he studied were, number 1, seed shape, seed color, flower position, pod shape, pod color, right? And then stem height. Now note down how many characters did he study related to the seed. So how many characters were related to the seed? There were 2 characters related to the seed. How many characters were related to flower? 2. One was color and second was position. Then how many characters were related to pod, shape and color? Again 2. And stem height. Please tell me that all these points are clear to all. Is that clear? So he studied total 7 characters. Now, out of these 7 characters, guys, he observed that there were 7 characters which occurred more in the population. So why do we call 7 characters as dominant? Dominant characters are basically which appear more in the population. They appear more in population. Gradually we'll understand why do dominant characters appear more. We'll see it scientifically, appear more in population. So hence dominant. And recessive occurs very less, right? So now see here, he saw that out of seed shape, round was dominant and wrinkled was recessive, right? Then in seed color, yellow is dominant over green color, flower color, violet is dominant over white, full shape of pod is dominant over constricted, pod color, green is dominant over yellow, flower position, axial is dominant. Axial is the space between the leaf and the stem. So see, so this is the stem, this is the leaf, and this from the axial the flower is arising. And if the tip is changing into the flower, this is called terminal. Now terminal is a recessive trait. Now you can directly get a very simple question: which of the following set of traits are dominant in nature, or following sets are recessive in nature? Is that clear? So you should know which ones are dominant, which ones are recessive. And stem height, tall is dominant over dwarf. Now the doubt can arise when you have to compare out of seed color. Now see, in seed color and pod color, right? So how do you remember that which one is dominant? So PG, paying guest, right? So PG, and then opposite is true for the seed. So in pods, guys, green is dominant, and in seeds, yellow is dominant. Is that clear? Is that clear to all? So will you be able to mark which are the characters, 7 characters that he studied, and which are the dominant ones and which are the recessive traits? Please tell me these points are clear to all, right? Now, moving on to, see, these are the few lines which I wanted to read with you. See, Mendel conducted some artificial pollination or cross-pollination experiments using several true breeding pea lines. We'll understand what are true breeding. True breeding are basically homozygous. They can be dominant as well as recessive. Both homozygous and, dominant and recessive of homozygous nature are called true. Is that clear? So, a true breeding is the one having undergone. And how is homozygosity obtained? By undergoing continuous self-pollination. So if you keep on performing self-pollination, it will lead to homozygosity. Now that you have to remember for sexual reproduction in angiosperms also. If you will keep performing self-pollination in plants, it will lead to homozygosity. Now there is both plus and minus related to it. So there is an advantage as well, there is a disadvantage as well. Advantage is that the homozygosity of positive traits will ensure that that positive trait is passed on from one generation to another. But similarly, the homozygosity of not so good genes will ensure that not so good character is also exhibited out, which was otherwise not visible, that also gets exposed. Is that clear? So it shows the stable trait inheritance and expression of that for several generations. Now see here, Mendel selected 14 breeding pea plants, 14 true breeding. What do we understand true breeding? Homozygous. We'll see that. And which were similar except for one character in the contrasting trait. And these are the contrasting traits. So these are the contrasting traits. They are contrasting to each other that he has studied. Is that clear? I'll come on to the crosses now. See, these are the cross pollinations that he is doing. Artificial hybridization. Why do we call it artificial hybridization? Hybridization means when we are crossing two different plants. Artificial because human involvement is there. Mendel is performing those. Mendel is choosing. So when humans interfere, humans intervene, humans make a choice that which two plants get to pollinate each other or cross, that is artificial. And when there are two genetically different plants crossing each other, it is also known as cross pollination. Is that clear? So see, we know the steps of cross pollination though, but we will restudy it in the chapter of sexual reproduction. So first of all, see, this is the female plant he is choosing. So the plant which is chosen as a female plant is always, we always remove the anthers, and this step is known as emasculation. Is that clear? And then he has chosen the male parent and transferring the chosen pollen grains. This is known as artificial pollination, and therefore the hybridization is artificial hybridization. And then he gets to see the overall results. Is that clear? Now there is this question: if a plant is unisexual, give me an answer. Although I'll be discussing it in sexual reproduction also, but because this point has come up here, you let me know if a flower is unisexual, which step will be avoided, which is mentioned here. So if the flower is, hashtag, if the flower is already a female flower, let's say if the flower which has been chosen as a female doesn't have stamens, so if flower is unisexual, therefore then what do we do? Then emasculation is, this step is skipped. Emasculation not done. But that is not the case with P, because P is a bisexual flower. Are we clear till here? Yes? Alright. Now, coming on to what are the reasons of Mendel's success and failure, we'll come on to that. But before now, we see the success and failure reasons, we need to understand what has Mendel contributed for genetics. So he is known as the father of genetics, right? He is known as the father of traditional genetics. Modern genetics is different, but father of genetics. Why? Because he laid the path, right? So on pea plant, we have seen he has studied a lot for 7 years. He is given 7 years for just pea plant. Imagine the love. And now, after these 7 years, he was able to conclude 4 different things. One, that was law of paired factors, right? Law of paired factors. What does law of paired factors say? Law of paired factors say that every character, one character is controlled by a pair of genes, a pair of factors. He had no idea. See, this movie is going to, play in the background. Mendel is of that time where there is no idea of DNA or such things. Are you getting that? So he had no such idea. And now he said that, okay, he's seen 7 characters. He says flower color is either purple or white. Plant is either tall or it is dwarf. The pod color is either green or yellow. So in every character, he is only able to see 2 contrasting traits. So what did he conclude? He concluded that one character is being controlled by a pair of factors. Later on, it was a different scientist who actually found out that the factor that Mendel was talking about is nothing but a gene. It is a gene, or we can call it alleles. Are you getting that? The alternate forms will be known as alleles. Is that clear? Then later on, the second conclusion that he gave was law of dominance. Law of dominance. Now see, for example, here I can say that for plant height was controlled by two factors. It is capital T or it is small t. Are you getting it? Then law of dominance. What does law of dominance says? Law of dominance says that in the heterozygous state, see, in the heterozygous state of genotype, let's say when the plant is, when the genotype is this, in that case, one of the allele will dominate over the other. So there will be one allele which will be always able to express itself. Now that which will always express itself even in the presence of a different allele, that one is a dominant one. And the other one which is not able to express itself in the presence of the dominant, that one is called recessive. See, I always take this example. So if there are two people who are saying that the color of the room should be, let's say, white. If both the persons are saying the same thing, we will never understand who is dominating over the other because both have the same opinion. It is always when there is a difference of opinion that you understand whose opinion matters more, whose opinion has been taken into consideration. That person is called the dominating one. Yes or no? So similarly, so law of dominance can be observed in the heterozygous state. It can neither be studied in this situation nor it can be studied in this situation because both have the same expression. Are we able to understand? Please let me know. Yes? So law of dominance came up. Now, then he gave us a third law, that is law of segregation. Right? Law of segregation. Law of segregation, by the way, is also known as law of purity of gametes. Purity of gametes. See, so what does this law say? This law says that whenever there is a gamete formation, see, we have read human reproduction, we know the concept of meiosis, right? So even he did. So he said that during gamete formation, because meiosis takes place in diploid parents, right? And during gamete formation, when meiosis occurs, so each gamete will get only one of the factors out of the pair. So why do we call it purity of gametes? Because each gamete will have only one type of character, one type of trait, sorry. It will have factor for only one type of trait. So it is pure. It is either carrying an allele for or a factor for tallness, or it carries the allele for dwarfness. So there is no khichdi cooking up here. Is that clear? So there is, during gamete formation, meiosis takes place and each gamete will have only one of the factors. So if the individual is this, let's say, if this is the parent, then there will be two types of gametes, one carrying capital T and the other carrying small t, both in case of sperm formation and in case of egg formation. Are we clear till here? Please let me know. Are we clear here? Yes? All right. Now then there came next law and the last law, that is law of independent assortment. Now I'll come to this particular law in a while, and first we'll discuss these three laws because these three laws were concluded after he did this first cross, that is monohybrid cross. Now what is monohybrid cross? In monohybrid cross you study, that is inheritance of one gene. Inheritance of one gene. That means only one character under study. Inheritance of one gene, and one gene controls only one character as per Mendel. Remember few things. So we are setting up the stage for all the new findings also, which will happen post Mendelian era, right? So according to Mendel, one gene will control one character only. Just remember these things. Only one character under study. Because as I said, according to Mendel, one gene will control only one character. Remember these things. I will be coming back to that. Now, See, we have just read that Mendel chose 14 true breeding plants. True breeding means homozygous. So every story begins with the parents, and parents have to be true breeding. Parents are true breeding. That means homozygous. Parents are always chosen as homozygous, right? So one parent is taken as dominant and the other parent chosen has a recessive trait. For example, let's say I am choosing plant height as a character here. Okay. So in that case, the dominant trait would be tall and the recessive trait would be dwarf. So I'm taking one tall plant and the other as a dwarf parent, right? Now, according to Mendel, because it is homozygous, so what is going to be the genotype here? That is the genetic composition. It will be TT here and small t, small t. Now, this is homozygosity or purity. Do you understand what pure lines mean? Now, how do parents reproduce? By the production of gametes. Now, at the time of gamete formation, see, there is only one law that is going to apply always. Okay, as the time will progress, as we go into modern world, technology improves and new findings happen, you will see that there are anomalies to all the Mendelian things, all the Mendelian principles, except for one principle that he gave, one law that he gave, that is law of segregation. This law of segregation has to apply. There is no exception to that. Is that clear? Now see, see, hashtag like hashtag yes, until and unless there is some deformity or there is disturbance in the meiosis, we will see that disturbance also happens sometimes, but normal situation, law of segregation will always apply. So when law of segregation applies, guys, what happens? One gamete will get only one type of the allele or the factor, the other will also get similar because the other is also of the same nature, and similarly here also it will form this. And now we know when the first generation is formed, F1 generation is formed, filial 1, so it gives us capital T, small t. Now 100% generation, F1 generation would be tall in nature. So do you see that F1 generation resembles, see carefully, it resembles the dominant parent. Dominant parent is tall, this is also 100% tall, but only phenotypically, not genotypically. Yes or no? Yes or no? So 100% tall generation will be formed. Is that clear to all? Now, if we go further and get this F2 generation, so must know that F2 generation is always obtained by selfing. Now, what do we understand by selfing, guys? That means on self-pollination. On self-pollination. Now, that means we have to cross capital T small t with capital T small t. Is that clear? Actually, it was after F1 generation that he gave the law of dominance, because I told you dominance, who is dominating will be understood when two have different opinion. Is that clear? Yes. So only this one was able to express itself. Now, let's talk about the F2 generation here. How did he form the F2 generation? So in F2 generation, we are going to form the gametes again. See, gametes. So here, according to the law of segregation, capital T small t will segregate, and similarly here also capital T and small t will segregate. So gametes will always be formed like this. So in F2 generation, I'll write down the gametes of one parent on one side and other parent on the other side, right? So it was Reginald Pinnet. It was the name of a British scientist who gave us this way of finding out all the possibilities. So he said, make this checkerboard to get the possibilities. So this is how, these are the possibilities in the F2 generation, right? So this will be tall, this will be tall, this will be tall, and this will be dwarf. So in F2 generation, see the conclusions here. In F2 generation, the phenotypic ratio. What are the two different types of phenotypes that you can see? Phenotype is the expressed trait. See, the two traits possible are either tall or dwarf, right? Now, how many are tall? 3 out of 4. or just write down 3. And how many dwarf? It is 1, right? So in F2 generation, guys, in F2 generation, the phenotypic ratio is always 3:1. And if I go for the genotypic ratio, genotype is the genetic composition, genetic composition. So what is the what are the different genotypes? We have homozygous dominant, we have heterozygous, and we have homozygous recessive. So what do we get? How many capital T? 1. How many capital T small t? 2. And small t 1. So this is the conclusion. This is the conclusion of monohybrid cross. Are we all clear till here? Yes. Now, I have used these terms multiple times till now. So I hope these points are clear to all. So what is, see, what Mendel called as allele or Mendel called it factor was later known as allele. And what is allele? Alleles are basically the alternate forms of a gene. It is the alternate form of a gene. So if a gene is in the dominant state, it is expressed as capital letter, and if it is in the recessive trait, it is expressed in the small letter of the dominant trait, the first letter of the dominant trait. Then gene, right, it is a part of DNA responsible for a character. So what, according to Mendel, that is a part of DNA, or say one gene is that part which controls one character. What is a genotype? It is the genetic composition. Now this is not visible, right? So what is a character? It is any feature under study. Plant height, eye color, nose shape, these are all feature under study. Is that clear? Now those features can be of different types. So different options, different possibilities, or different expressions of the character are known as traits. Now, out of those many expressions which are possible, the one which is present in the individual, is known as phenotype. So it is these are the different expressions of the character possible, and this is the expressed one. So the expressed trait is known as phenotype. Please tell me that these points are clear to all. Please tell me, guys, that these points are clear to all. Physically appeared one. Yes, absolutely. Absolutely. The one which is physically present, that one is known as phenotype. Is it clear? What is the chromosome number for each Sankalp? Chromosomes, the concept of chromosomes is not here yet. We'll come on to chromosomes also. Are these points clear, everyone? Now, guys, moving on to the next. See here. Now, Mendel gave us dihybrid cross. See, from the monohybrid cross till here, from monohybrid cross, there are just 3 laws that will be clear. Monohybrid cross gave us 3 laws. Law of paired factors, yes. Law of dominance, right? And law of segregation. These are the only 3 laws which were concluded after monohybrid cross. But when he did the dihybrid cross, then 4th law will also come up. So law of independent assortment will not be studied through monohybrid cross. That is also a question. So now we'll get to see a 4th law when he studies the dihybrid cross. What is dihybrid cross? This is inheritance of 2 genes. So 2 genes means 2 characters will be studied. So 2 characters under study. It can be any 2 characters under study. Is that clear? That is dihybrid cross. Now see here, what did Mendel do? Here? So he took, so he is studying seed shape here. So he is studying seed shape and seed color. So this is dihybrid. Is that clear? So one parent is taken dominant, so round shape and yellow color, and wrinkled shape and green seeds. So do you see the genotype here? And what will be the gametes like? So you'll take 1 R from here and 1 Y from here, and similar small r, small y. So F1 generation will be round and yellow. Again, the F1 generation resembles the parent, dominant parent, but phenotypically. And on selfing, we'll get the F2 generation. So F2 generation is always obtained on selfing. So you write down the gametes here and write down the gametes here. Now see, you know that during gamete formation, one law is always and always applicable, that is law of segregation. Yes. So out of a pair of a gene, only one will be passed on to the gamete, right? But now when we are considering two genes simultaneously, there will be another law applicable here, and that is law of independent assortment. Law of independent assortment. See here, what does independent assortment say? Just think about your classrooms. You all have your own friends. You have chosen the person you want to sit with, right? And you have always chosen a seat that you want to sit on, right? Now see, so independent assortment says whenever there are more than one genes involved, let's say we have capital A, small a, capital B, small b, right? So according to law of segregation, one gamete will get capital A, another gamete will get small a, right? Now similarly, from capital B and small b, one should go with one, other should go with the other. Now here Mendel said that it is not mandatory that, see, it is not mandatory that capital A will sit with capital B only. Capital A can sit with small b also. There are there is equal probability of capital A going with capital B and capital A going with small b, right? Similarly, small a can go with capital B as well as it can go with small b also. Therefore, we will not get just two types of gametes here. We will get four types of gametes. So capital A going with capital B, or it is going with small b, and similarly small a going with capital B and small a going with small b. So this is law of independent assortment. So this law of independent assortment is only possible when we are considering two genes, at least two genes simultaneously. Please tell me that these points are clear to all. Are we clear? Equal chance of distribution. Very good, my universe. Right? So when the two genes are getting distributed during the gamete formation, there are equal chances that any factor can go with any other factor of the different gene. Is this point clear? So according to, I'm going to give you some formula here without, where you don't have to make different gametes, and you can quickly find out how many different types of gametes will be formed. We will be coming on to that. So when we did this combination, so in the phenotype, in the F2 generation, the phenotypic ratio came out to be 9:3:3:1. Is that clear? So when he took round and yellow parents and another was wrinkled and green, what was the F2 generation? Now see this very carefully. In F2 generation, guys, in F2 generation of dihybrid cross, he saw four hybrid cross. Number one, he saw four phenotypes. So first was round and yellow. Then it was round and green. Then he saw wrinkled and yellow, and he saw wrinkled and green. Is it clear? And how many he found out? So 9, 3, 3 and 1. Out of how many? 16, right? If we check the number of individuals formed, it is 16. So out of 16, 9, 3, 3 and 1. Is that clear? Now see here, what were the parents like? Round and yellow and wrinkled and green. So can we say that round and yellow and wrinkled and green, these are the parental types? Yes or no? These two are the parental types. These two are parental types. Yes or no? So how many parental types have been formed? How many parental types? So 9 plus 1, that is 10 out of 16. So out of 16, how many are parental types? 10 out of 16. Yes. And then can you find out the percentage quickly? Can you find out the percentage? 10 out of 16 into 100. Find it out. And then these are the two different combinations that have been formed. So I'll call these as recombinants. I'll call these as recombinants. How many recombinants? 3 plus 3. That is 6. Out of 16, it is just 6 which are recombinant types. Quickly find out the percentage. Yes, please. Can you tell me the percentage here? Can you tell me the percentage? Very good, Shobha. So here we get 62.5% and here we get 37.5%. Am I right or not? So can we say that this is the parental frequency? This is the parental frequency and this one is the recombinant frequency. These results are going to be very, very important when we study linkage. Please note it down. Yes, guys, please note it down. Yes? Is that clear to all? Is that clear to all? Right? So this is the conclusion of F2 generation. Now remember, in dihybrid cross, also we can study the monohybrid ratio. For example, we can derive the monohybrid ratio also here. How? So if we have to derive the monohybrid ratio here, so what do? I'll just choose one character at a time. So one character would be, let's say, seed shape. So round is to wrinkled. How many round are here? So 9 and 3. These two are round. So total 9, 10, 11, 12. So how many wrinkled are there? 4. So that will still come out to be 3:1. So the monohybrid ratio is also constant. Is that clear? So if we want to derive it, we can derive it. And similarly, if I want to calculate for seed color, how many yellow? 9 plus 3, 12 again. How many green? Again 4. So 12:4. So even if it is dihybrid or trihybrid, you will always, if you extract the monohybrid ratio, that will always remain the same. Please tell me that these points are clear to all. Yes, guys. Are we clear till here? Can we move on? Yes. So law of independent assortment is clear to everyone, right? So this law of independent assortment can only be studied in at least I'm saying at least 2 gene inheritance. It can be seen in 3 or 4 or 5, whatever, but at least there has to be 2 genes inheritance where we can see this happening. Is that clear to all? Now, guys, this slide is very, very important. Please take a screenshot also. And now let's see the formulae here. These formulae apply during Mendelian genetics and they will help us to quickly calculate things, right? So see here. So if I have to calculate the type of gametes formed. So type of gametes can be calculated by using the formula 2 raised to power n. Now see here, n is the number of heterozygous conditions. Is that clear? Then, for example, wait. Yeah, for example, see, number of heterozygous conditions. So I'm again saying this slide will help you to calculate things very quickly during the exam. So if I have to calculate the type of gametes, the formula is 2 raised to power n, where n is the number of heterozygous conditions. Let's quickly see. If it is this, if this is the genotype and I ask you how many different types of gametes are formed, what are you going to do? You're just going to quickly see how many heterozygous conditions. This is also heterozygous, this is also heterozygous. So the formula applies 2 raised to power 2. So there will be 4 different type of gametes formed. You can check. So let's say if there is a different condition, for example, capital A, capital B, small b. Now in this situation, how many number of gametes are formed? See here, 2 raised to power. How many heterozygous? This is homozygous, so I will not count it. This one is in the heterozygous state. So 2 raised to power 1. How many different type of gametes will be formed? 2. Now if I take a bigger genotype, let's say it is this. You can take any genotype. This formula applies everywhere. Is that clear? Now, see here, how do we calculate the number of gametes? So it is 2 raised to power n. Yes, and n is the heterozygous conditions. Is A in the heterozygous state? Yes. Is B in the heterozygous state? No. Is C in the heterozygous state? Yes. Is D in the heterozygous state? Yes. So how many heterozygous conditions are there? 2 raised to power 3. How many different type of gametes will be formed? 8. Solve, check and try. This formula will apply everywhere in calculating the type of gametes formed. Is that clear? Now, if guys we have to calculate number of phenotypes that are possible, so the formula would be for number of phenotypes, the formula is 2 raised to power n again, but now n is different here. So n is the number of gene pairs. So how many genes are involved? See here. So in all these conditions, this and this, how many genes are involved? 1 gene, 2 gene. So 2 raised to power 2 will be 4. Here also, how many gene pairs are involved? 2. Is that clear? So you will not calculate the heterozygous condition here. You will just see how many genes are present. For example, if I take this genotype, how many gene pairs? 1, 2, 3, 4. There are 4 gene pairs involved. Yes. So 2 raised to power 4. Can you calculate? 16. So 16 different phenotypes are possible. Now, if I have to calculate the number of genotypes possible. So how many number of genotypes, genetic compositions are possible? So for this we have guys this formula 3 raised to power n, and again n becomes the heterozygous conditions. So in this situation, heterozygous condition, the same one which I have used above. So these formula will help you calculate things very quickly during the exam. Please take a screenshot and let me know that these points are clear to you. Please let me know guys. Yes? All right. Now moving on to the next part, which is called a test cross. Now see here. Do you remember in the beginning I said that dominant trait is the one which expresses itself in which is found more in the population, right? Why is it found more in the population? Because the dominant trait can express itself in 2 situations, in homozygous dominant also, as well as in the heterozygous state also. Yes, in both the situations, let's say I'm talking about tallness, so this is also tall, this is also tall, right? And we know how many capital T are there? 1. How many this are there? 2. In a Punnett square, in a typical Punnett square, 1 out of 4, 2 out of 4. So total 3 out of 4 will be tall. Now you know why they are more in the population, because they are able to express in homozygous dominant situation as well as in the heterozygous. And we already understand now why recessive trait is in the lesser frequency, because it is only able to express itself during the homozygous recessive state, which is also just 1 out of 4. Is that clear? Right? Now, that means, now understand very carefully, that means if somebody tells you that the plant is dwarf, then you are always sure, then you should be always sure that the genotype of the dwarf plant is this. So we are always sure about the genotype of the recessive trait. Yes or no? Do we understand this? If I say the plant, so the flower color is white, we are always sure that what will be the what will be the genotype? Let's go back to this this one. Yes, here. Now watch carefully. So if somebody tells me, so first of all, what will be the allele for this round? It will be R, this will be yellow, Y, this will be V, this will be F, this will be G, A and T. So the allele is always chosen on the basis of the dominant trait. The first letter of the dominant trait is taken as a symbol for the allele. Now the recessive is expressed in the small letter, small r, small y. Is that clear? V, then small f, small g, small a and small t. What did I just say? We are always sure about the genotype of the recessive trait. Why? Because recessive trait can only express itself in homozygous recessive condition, there is no other way in which it will come out or get exposed in the population. So I am always sure about the genotype of wrinkled is this is this. We are sure about this. Are you clear? But I am not sure about the genotype of the dominant trait, because dominant trait can be in homozygous dominant condition also, as well as in the heterozygous state. I don't know whether this dominant is capital R or capital R small r. Are you getting that? Now, if you want to analyze, or if you want to find out if the dominant trait is because of the homozygous condition or heterozygous condition now, so that for that thing, guys, we have a very important genetic tool, and that genetic tool is called the test cross. So it is a very important genetic tool to determine the genotype of dominant trait. Is that clear? Because dominant trait can be because of homozygous condition as well as heterozygous condition. Please tell me, are you able to appreciate this point or not? Is that clear? So dominant trait is because of capital T or capital T small t. Please tell me if you are clear about this point and you are able to appreciate. Yes? Yes or no? This point and you are able to appreciate. Yes? Oh no, the voice is totally fine. The voice is totally fine. Now see here. So this is a genetic tool. So in what do we do basically in the test cross? So we cross the dominant individual whose genotype you want to determine. That dominant individual is crossed with homozygous recessive parent. So this is what we do in the test cross. Whether it is for one gene inheritance or two gene inheritance, you can do it for all, or tri-hybrid also, this works. Is that clear? So the tall individual will be crossed with the dwarf individual. Now we don't know. See, we are sure about the dwarf, that this is small t. We are not sure about this or this. So this is why we are doing this experiment. We are doing this cross. Is that clear? Now watch carefully, guys. See, in case, see, they have taken this example of violet flowers. Okay. Now, in case here, if the flower is capital W, that means in the homozygous condition, if the flower is in the, sorry, this is capital W, that is the flower is violet, but whether it is capital W or capital W small w, this is what you want to calculate here, right? Now, in that situation, we are crossing it with small w, here also small w, homozygous recessive. Clear? Now, see the result. If I cross this homozygous one with the homozygous recessive, I get all flowers violet, which is a dominant state. So, if you get all the dominant phenotype in the generation, that means parent was homozygous dominant. But if you see 50% dominant trait and 50% recessive trait, that means the parent was heterozygous. So if you get violet and white in the ratio 1:1, it could be any character. If you get both the traits, dominant as well as recessive, in 1:1 ratio or 50-50% ratio, then it means the parent was heterozygous. And if you get 100% dominant trait, 100% dominant phenotype, that means the parent was homozygous dominant. Is this point clear? Is this point clear? Please tell me. Yes, guys. Now, this can be done for dihybrid also. In case of dihybrid, you will get, if the parent is, let's say, in this condition, that is homozygous dominant, then you will get 100% dominant trait. Is that clear? And if the parent is in heterozygous condition for any two traits, then you will get ratio 1:1:1:1. So you should be able to identify if there is this ratio. They are usually taking test cross into consideration. Please tell me these points are clear. Please tell me these points are clear to all. Yes or no, guys? Yes or no? Is that clear? Yes? Perfect. Now moving on to the next part. Now we are going to enter the world where we are going to research Mendel's work, and we are going to find that of course he was right. We deserves to be known as the father of genetics. He laid the foundation of a new branch of biology. But then there are other things also. We'll see why Mendel was successful. See, Mendel was successful for many reasons. Number one, he was a lucky guy that he happened to choose a plant which just showed very contrasting traits. There was no intermediate character seen. So it is either violet or it is white. It is either tall or it is dwarf. It is either green or it is yellow. Are you getting that? There's no intermediate feature that was visible. So Mendel was lucky in that manner, right? He happened to choose that plant by chance. But then, and also he had a large sampling size. So the calculations that he did, that also concluded his ratios very well. But then there are some failure reasons. Being a mathematician, he had that habit of converting everything into a formula. Now just imagine, a plus b is equal to a square plus b square plus 2ab. When you hear this formula, nobody will think about genetics for the first time, right? We know that this is a genetics ka formula as well. But for a lay person who knows, or even for a mathematician, it is just a mathematics ka formula and not a formula of genetics. So that became a weakling also for his successful career actually. And he had just compiled everything and put up in his like trunk, and he died. So later on there were other 3 scientists who rediscovered Mendel's work, restudied, re-experimented, and gave him the due credit. Is that clear? Okay, so Mendel was actually lucky. Now otherwise, it is not mandatory that people who research your work, they'll give you the due credit, but they were good people who did that. There were 3 scientists. Let me know the answer. Let me know the answer, the name of those 3 scientists who rediscovered Mendel's work. Let me know in the chat. And those of you who will be watching it later, you have to leave the comment also. Name the 3 scientists who rediscovered Mendel's work. Now, I am coming into the post-Mendelian era, where there were some exceptions found around the Mendelian laws. Now see, Mendel gave us law of paired factors, but then we found a concept of multiple allelism. What is it? We'll see. Mendel gave law of dominance. We found out incomplete dominance and codominance. He gave law of independent assortment. Linkage came up. Is that clear? So all these things in pink are the exceptional phenomena. These are exceptions to the laws or the things said by Mendel. Then Mendel also said one gene controls one character. Remember, one gene will control one character. We will come across a phenomena of pleiotropy, where we will see one gene controlling multiple characters. Then he also said one character will be controlled by one gene. We'll come across a phenomena of polygenic inheritance. So you must know that these are exceptions to Mendelian laws. Is that clear? Yes or no? Right? All right, guys. Now, moving on. And before I move on, guys, I am going to tell you. So, by the way, June has started. 21st June is not very far. So, we have to start practicing our questions. So, number 1, there is All India Mock Test Series going on every thrice a week. Is that clear? So, we have to practice questions. So All India Mock Test Series is number 1, and we have to practice questions on daily basis, guys. So for daily basis practice, we are conducting special classes absolutely free on Unacademy Learners app. Just the description, it will be given in the description box. You can follow me there, and every 2 p.m. every day 2 p.m. I'm conducting a special class where we are practicing the questions. Also, I have started with NCERT exemplar based questions. I've completed 1 unit today. I'll be doing that. I'll be practicing, solving questions from unit 2, exemplar based questions. So don't miss these special classes, don't miss the practice sessions. They are very, very crucial for your preparation. Is that clear? All right. So moving on now. So 2 p.m. is the time that you have to remember. Now see. So first of all, for law of dominance, where Mendel said one will always dominate over the other. But we got this concept of incomplete dominance, where when it is in a heterozygous state. See, when was law of dominance concluded? When it was a heterozygous state. I told you, who is dominant out of the two persons is always concluded when both of them have a different opinion. One says red, other says white. What will happen? Red. It means red was dominant. But we found out that in case of flower color, now see, you have to remember these points. Flower color in Snapdragon. These are the examples. So the examples of incomplete dominance can be studied in example 1. Flower color in Snapdragon, also known as dog flower. The scientific name is antirrhinium. Is that rhinium? So that shows incomplete dominance. And second, flower color in Mirabilis jalapa. Mirabilis jalapa. This is also known as a 4 o'clock plant. Is it clear? So in both these plants, guys, we get to see that when we cross a red flower, when we cross a red flower, red is because of homozygous state, capital R, with the white flower, which is because of small r. So as per Mendelian genetics, what should have happened? Capital R, small r should have been red, right? Now this is Mendelian, but we did not get this. We did not get red. Instead, we got 100% pink. We got 100% pink. So it was neither capital R nor small r. Both of them were not able, neither of them was able to dominate. Otherwise, according to Mendel, one should have been able to express itself completely, and that would have been concluded as the dominant one. But no, we discovered that in certain cases, like in case of Snapdragon and Mirabilis, when it is a heterozygous state, neither of them is able to dominate completely. Rather, we get an intermediate feature. We get an intermediate feature. So whenever there is an intermediate feature, not red, not white, it became a new intermediate, that is pink in between. That is a case of incomplete dominance. Is that clear? Now F2 generation, we know, is obtained always by selfing. So when we did selfing, so pink and pink were crossed, we got, see, this ratio is important, we got 1 red, 2 pinks and 1 white. And what was Mendelian ratio? It was either red or white. 3 red, 1 white. But he got 1 red, 2 pink and 1 white. Do you see the deviation here? Do you see the deviation? So Mendelian ratio would have been 3:1. But what did we get here? This is the deviation, not 3:1, 1:2:1. And similarly, the genotypic ratio, well, that remains the same. That remains the same. That is 1:2:1. So that means here we are getting both genotypic ratio and phenotypic ratio same in case of incomplete dominance. Are we clear? Please tell me these points are clear to all. Are we clear? Yes. So these points are clear to all. Yes. Both phenotypic and genotypic ratio will be 1:2:1. Then we come across another phenomena, that is codominance. See, in incomplete, what happens? It's neither red nor white, it is pink. Had it been codominance, codominance means both the dominant are able to express themselves simultaneously. See, what is the difference? Here, neither of the allele expresses completely when in the heterozygous state. In heterozygous state or condition, right? Neither of the allele. But in codominance, both the dominant alleles are able to express simultaneously. So I want the walls white, you want the walls black, then what will happen? Half white, half black. That is codominance. Is that clear? Now see here. So we get to study this codominance in a blood group inheritance. So also in blood group inheritance, guys, in blood group inheritance, we see human blood group inheritance. We see that the ABO blood group is controlled by one gene which is called I gene. Is that clear? But this I gene, instead of having two alleles, law of paired factors, now we are going to see an exception to law of paired factors. Now this gene has three alleles. So it is not a pair here anymore. So this gene has how many alternate factors? Sorry, alternate forms, three alleles. So it is IA, IB and small I. Is that clear? Now these alleles occur in population, not in an individual, occur in population. An individual can only have any two of these. So see here, an individual can have only any two of these. So which two alleles out of these will be present in you, that depends upon your parents. Is that clear? So any two will occur, but in population we get to see three alleles. This phenomenon is called multiple allelism. One gene, multiple forms. Is that clear? So that is a case with ABO blood group controlled by three alleles. For example, Drosophila eye color, that will be controlled by 14 different alleles. We don't have to go there, but just telling you one gene can have multiple forms as well. Is it clear? Now see, so this is multiple allelism. Now say one, so here I'm going to discuss. So see here, wait, so any two will combine. It can be IA, homozygous, IA, small I. Now this is recessive. These both are dominant, right? That is why we are writing it in capital. Now see here, it can be this homozygous and it can be heterozygous this. It can be IA, IB and it can be small I, small I. Now see here, this will be A blood group. These two will be B blood group, this will be AB blood group and this one will be O blood group. Now watch. These two follow simple law of dominance. Homozygous state A, homozygous A, heterozygous form A. Is that clear? Homozygous B, heterozygous B. Now if you are A, and in order to know whether you are homozygous A or heterozygous A, which is the genetic tool we have to identify the genotype of the dominant trait, that is test cross. So if you want to find out if you are A and which A you are, homozygous or heterozygous, you will have to perform the test cross. Are these points clear? Now see here, this one. Do you see, in this case there are two dominant alleles present together. So IA small and IB both are dominant and both are able to express themselves simultaneously. What will your RBC look like in this case? So this is your RBC. Your RBC will have A glycoproteins as well as B. A as well as B. So both alleles are able to express themselves simultaneously. This is codominance. So AB blood group is a case of codominance. Are these points clear? Yes. And then O blood group, again law of dominance. So the recessive trait will always be in the homozygous recessive state. Please tell me these points are clear. So only two types of blood groups, the individuals of two types of blood groups should be sure about their genotype. If you are AB, you are showing this codominance, you should be sure that you are IA, IB. And if you are a blood group O individual like me, then we are also sure about a genotype that we have small I, small I. Is that clear? So our RBC looks like this. It is naked. So there is neither A nor B. We are not considering RH factor here, just to mention. Is that point clear to all? Please guys, let me know. Haswant, you are very, very right. You're right. Is that clear to all? Yes. So now, what will be the genotype of the offspring? That will entirely depend upon the genotype of the parents. Can we cross and find out? Yes. Now, there are other few things that you must know. So in that case, guys, in case of human blood group, in human ABO blood group, we must know, we must know that there is I gene controlling it, and this I gene has 3 alleles. Number 1, 4 different phenotypes, and how many? 6 genotypes possible. So these are the lines written in your NCERT. So let's watch again. So there are 3 alleles. There are 4 phenotypes. Phenotype is the type of blood group. These are the phenotypes, right? It can be A, B, AB or O, 4, and 6 genotypes are possible. 1, 2, 3, 4, 5, 6. These are the 6 genotypes possible. Please let me know that these points are clear to you. And in human blood group inheritance, you can study 2 phenomena. One is multiple allelism, right? And you can also study codominance. These are the exceptional cases to Mendelian inheritance. Of course, Mendelian inheritance also applies here in other A and B blood groups. Is that clear? Now, coming on to second exceptional case. Sorry, fourth. We have talked about multiple allelism, incomplete dominance, codominance. Now coming on to pleiotropism. See, Mendel, what did Mendel say? According to Mendel, one gene controls one character, right? One gene controls one character. Now you see the exception. Exception is pleiotropy, where one gene controls many, it controls many characters, multiple characters. Is that clear? One gene is going to control multiple characters, out of which these multiple, one will be primary, other characters will be secondary. Now we have 3 examples here. First example is sickle cell anemia, second is phenylketonuria, and third I'm going to study B gene inheritance. Now let's talk about sickle cell anemia. So basically, why does sickle cell anemia happen? Now see, in sickle cell anemia, what happens? Where your mRNA should have GAG, instead of this, A is replaced with U. So this is a substitution mutation, type of mutation. So where single base pair is replaced, A is replaced with what? A is replaced with U. Do we see this? Yes. Now this one difference has led to a lot of, it has led to a big situation here. So see, GAG is responsible for glutamic acid. When we'll study the genetic code, GAG is responsible for glutamic acid, while GUG is responsible for valine. Is that clear? So what happens in sickle cell anemia? So we know we have 2 alpha chains and 2 beta chains, right? And in beta chain, guys, in beta chain, I'm discussing the beta chain here. So the amino acid numbers 6, 4, 5 and 6. So the 6th amino acid and so on. 6th amino acid should be glutamic acid. It should be glutamic acid. It should be GAG coding for glutamic acid. But if this is replaced by valine, what will happen here? Now, why will it be replaced? Because GAG is substituted by GUG. A is substituted by uracil, right? Now, in that case, what will happen? The beta chain will be destroyed. The structure of beta chain will destroy. Beta chain structure changes, the hemoglobin structure changes. Now, if the structure of hemoglobin changes, who is the carrier of hemoglobin? It is RBC. Now, the structure of RBC also changes. So, beta chain changes, then it leads to change in the hemoglobin structure. If hemoglobin structure changes, it also changes the RBC shape. RBC shape also changes. What happens? So from biconcave structure, so this is the typical structure of RBC, biconcave, it becomes sickle shape. Remember, a saw-like structure which is used by the farmers to cut the grass. So it becomes sickle shape, the sickle shaped RBCs. Now this is the primary character. This is the primary character. So I told you here one gene is going to control multiple characters. So primary character is that this one substitution changes the RBC shape, but the change in the RBC, this shape, it will cause secondary effects as well. So there are primary effects and then there are secondary effects. What are the secondary effects? This shape, when it is sickle shape, these RBCs are going to entangle with each other, form clots, form blockages in the capillaries. One, there are blockages. You know the repercussions of blockages in capillaries. It can lead to hemorrhages or other serious conditions. Then the oxygen transferring capacity, oxygen carrying capacity of hemoglobin as well as of RBC also gets changed. Less oxygen transport means breathlessness, bluishness on the skin, less energy, fatigue, lethargy, all that is a secondary effect of sickle cell anemia. Are you getting that? So here we see that one gene is having more than one effect, primary effect and secondary effects. Do we understand, guys? Do we understand what is this? Yes? Okay. Now similarly we have second example of phenylketonuria. Now see what happens in phenylketonuria. There is this an amino acid phenylalanine. Phenylalanine. This amino acid gets converted into another amino acid called tyrosine in the presence of an enzyme called phenylalanine hydroxylase. Phenylalanine hydroxylase. This is the enzyme. Now we know that enzymes are proteins and proteins are formed by genes. Now what happens in phenylketonuria? The gene which is producing this enzyme becomes non-functional. No gene, no enzyme. What will happen? If there is no enzyme, this phenylalanine will not change into tyrosine. Rather, phenylalanine accumulates in the body. Phenylalanine, it will accumulate in the body. Accumulates. Yes. So primary effect is no gene, no enzyme, phenylalanine is not converted, hence it is accumulated in the blood. What happens then? When phenylalanine is accumulated, it leads to mental retardation. It leads to mental retardation. Yes. It leads to kidney failure because kidney has to filter it. So kidney failure. If there is something in excess in your blood, it will showcase on your skin. So skin pigmentation. Is that clear? So 1, 2 and 3. All these are the secondary effects. This one is the primary effect. Do we understand? So one gene which is forming enzyme forming gene, this one gene is having multiple effects, primary effect, secondary effects. This phenomena is called pleiotropy. Do we understand this? Please tell me. Yes or no? Is that clear to all? Is that clear to all? Yes? All right. Now I'm going to talk about this B gene inheritance. Let's take it on the next page. So B gene inheritance, please understand it carefully. This is not very clearly mentioned. It is very well given in your book, but not very clearly mentioned. So I'm going to explain it separately. Now this B gene is able to control two things. One is the shape, seed shape, and second is the size of starch grains. Is that clear? So do we see multiple effects? One gene, two effects, that is pleiotropy, hence proved. My first point is clear here. Yes? Now, let's say this B gene can occur in, of course, 3 different conditions. These are the genotypes. These are the 3 genotypes possible. Now, if we have this and this, that means homozygous and heterozygous condition, in both these cases, the shape of the seed will be round. Only in this homozygous recessive state, the shape of the seed will be wrinkled. Yes? But now, talking about size of starch grains, see here, when it is homozygous dominant, the starch grain is very large in size. If it is homozygous recessive, sorry, heterozygous, it is intermediate. While when it is homozygous recessive, they are small sized. What type of inheritance do you see here? Please let me know in the chat. What type of inheritance do you see here, guys, when we are discussing the size of starch grains? Please tell me. Incomplete dominance, yes or no? Yes or no? So in B gene inheritance, you are able to see pleiotropy because it is controlling two characters, seed shape and size of starch grains. Here you observe typical Mendelian law of dominance, right? While when we study the starch grain size, we are able to observe incomplete dominance. Please tell me. So now you can get question that if the seed is round with intermediate starch grains, which of the following is the possible genotype? You should be able to crisscross and find out. Please let me know if these points are clear to everyone. Very good, Vishwa and Siddesh. Very good. Are we clear? Are we clear here? Now, moving on to another exception. See, Mendel said, according to Mendel, let's discuss Mendel. Mendel gave us a pathway. Mendel laid the foundation, right? Now, according to Mendel, guys, one character will be controlled by only one gene, right? But in polygenic inheritance, we see that one character will be controlled by multiple genes, controlled by many genes. Now, this is not a usual case. This is, of course, an exception, right? For example, human skin color, in human intelligence, human height, human eye color. These all characters, they are not controlled by just one gene. Because see, if there is just single gene inheritance, there are only two phenotypes possible. We have seen the formula also. Number of phenotypes can be calculated as. You must know that slide. Is that clear? But when there are multiple genes, so if, let's say, if there would be just one gene controlling human skin color, there will be only two types of heights possible, either a particular 6 feet or other 5 feet. That's it. Or maybe even lesser than that, or dwarfs, right? Either 6 feet or just dwarfs. Possible. So when there is just one gene inheritance, and that too Mendelian way following, there are just two phenotypes possible. Yes, but that's not the case here. In polygenic inheritance, one character is controlled by many genes. Hence, this is also known as multiple genes. See, I am not talking about multiple alleles here. I'm talking about multiple genes. Another point here, every gene adds to the effect of other genes. So it is also called additive genes or additive inheritance. Is that clear? In this, guys, it is not the quality of the gene that is mattering. It is the quantity of the genes that will matter. It is the quantitative inheritance. It is the quantitative inheritance. Please tell me these points are clear. So that is why you don't see just two extremes. You see a range of phenotypes here. There are different ranges of height. There is different ranges of intelligence in height, in skin color, in eye color. Is that clear? That is always possible when there is polygenic inheritance. Are we clear to all? Is this point clear to all? So if we talk about human skin color, so see, there are three genes, A, B and C. And now because we are diploid, we'll have 2 copies of each, right? It could be any. So ultimately there will be 6 alleles. Now these 6 alleles can be in any form. So now here in 6 alleles, guys, which 6 alleles will be present in an individual? Of course, your genetics, your parental combination is going to matter here. But now see, it can be all the 6 dominant. All 6 dominant. It could be any 5 dominant. It doesn't matter which ones are dominant. Is that clear? For example, it can be AA, BB, C and small c, or it can be this, or it can be this one. So in that case, in this case, any 5 can be dominant. Is that clear? Here all are dominant. Are you getting that? So any 4 are dominant. Similarly, any 3 dominant, any 2 dominant, any 1 dominant, and when there is no dominant gene, that means all recessive. So here it is not the quantity that will matter. Here it is not the quality that matters, that which one is dominant. It is the quantity that is going to matter. Do we see? Do we understand this point? Yes. Please let me know. Do we understand this point? It is the number of alleles which are dominant, right? Now, in human skin color, these genes, they contribute to melanin production. They contribute to melanin production. So, more the number of dominant alleles, more darker is the color. More is the melanin and darker the color. Is that point clear? For example, see here now. If all the 6 are dominant, maximum melanin will be formed, color will be very dark. We also call it negroid. We call it negroid. When any 5 are dominant, color will be dark. 4 dominant, fairly dark. Is that clear? Then 3 dominant, it will be intermediate color now, or mulatto, Is that clear? Then 2 dominant, now color will start going for the fairness side. So it will be fairly light. Dominant, it will be light. And no dominant would be very light. That means no melanin at all. No melanin at all. Neither in hair, nor in lashes, nor in brows. There is no melanin at all. So such condition is known as albinism. So guys, we are only sure about the genotype of 2 types of individuals. See here, we are only sure about those which don't have melanin. So what will be the genotype here? The genotype would be small a, small b, small b, small c, no dominant at all. And also we are sure about the negroid one. In that case also we are sure all the 6 are dominant. We are not sure about rest of them. Which one is dominant, we don't know. How many dominant, we can tell. Is that clear? So 3 genes, A, B, C, 6 alleles, any, and they, which one is dominant recessive, that depends upon your genetics. And then 6 alleles will always give you 7 phenotypes. Is that clear? Now, if I cross a negroid, with an albino, that means capital A, capital B, capital B, capital C is crossed with small a, small b, small c, then if I get the F2 generation in the exact same manner, it is a trihybrid cross. There is 3 gene inheritance in that case, right? So what would be the phenotypic ratio here? The phenotypic ratio would be 1:6:15:20:15:6:1. So that means one will be negroid, other will be dark, this will be fairly dark, this will be intermediate, this will be fairly light, this will be light, and this one will be albino. This is a fixed ratio. Are we clear, guys? Please let me know. Are we clear? Yes or no? Are we clear, everyone? Right? Okay, guys. Now moving on to, now this slide is again very important. These formula will apply only in case of polygenic inheritance. Now if you want to calculate, see here, if you want to calculate the number of phenotypes possible in polygenic inheritance, what is the formula? Formula is 2n plus 1, where n is the number of genes. N is the number of genes. For example, in human skin color, we have 3 genes, so you put it here. 2 into 3 plus 1, that will be 7. So did we get 7 phenotypes here? Also, the logic says we are calculating how many number of alleles are dominant. When all 6 dominant going up to 1 dominant. So, and one last condition when there is 0 dominant allele. So that is why it is always 2n plus 1. So this is the formula to calculate, and n is the number of genes involved. So if there are just 2 genes involved, so 2 genes can also show polygenic inheritance. In that case, it would be 2 into 2 plus 1. So that would be 5. Or you can say the number of phenotypes is equal to number of alleles plus 1. This can also be the formula. So how many alleles are there in human skin color? 6. 6 plus 1, you can go that way also. And how many alleles for 2 genes? 4. 4 plus 1, 5 phenotypes. So you have 2 formula to calculate the number of phenotypes in polygenic inheritance. Now we have number of genotypes. So number of genotypes, we have the same formula that we were using in Mendelian genetics, 3 raised to power n, where n is the number of genes, gene pairs actually. Is that clear? Number of gene pairs. Is that clear? So that is the number of genotypes. Then guys, we have formula to calculate the F2 phenotypic ratio. So in order to calculate the F2 phenotypic ratio, like we have calculated it here, 1:6:15. How did we calculate this? And what is the generic formula? So formula is 2 plus n raised to power n. Is that clear? So if there is just one gene involved and showing polygenic inheritance, that would be 2 plus 1 raised to power n. Is that clear? Usually you won't get this formula, you won't get to apply this formula, but this is the formula to calculate the phenotypic ratio. You just remember the phenotypic ratio for human skin inheritance. Is that clear? Please tell me these points are clear to all. And now, if I have to calculate, if I have to draw the graph here, how would the graph grow like? So see, here I am taking the phenotypic ratio on the y-axis, and on the x-axis I am taking the phenotypes. Is that clear? If I take the phenotypes, negroid, dark, fairly dark, intermediate, and so on. So I will see first the graph goes up till 20 and then the graph goes down. So in this case guys, that is polygenic inheritance, the shape of the graph is always bell shape. The shape of the graph is always bell shape. Is that clear to all? So first we see the increase, and then after the intermediate is reached, we see the fall, or we see the other side of the trait also in the same ratio. Are these points clear to you? So we get a bell-shaped graph. Yes or no? Please tell me that these points are clear to you all. And now guys, now we have another big situation coming up, post-Mendelian era. Now after Mendel, we have seen different concepts coming up. Now came the concept of chromosomes. So Mendel was just talking about factors. He didn't even know about genes. That term gene was also given later on by Bateson. Now came 2 people, Sutton and Boveri. Okay, these people studied the chromosomes and the presence of genes on chromosomes. So when they studied the presence of genes on chromosomes, they said chromosomes are the carriers of genes. What did they say? Chromosomes are the carriers of genes. Chromosomes, the carriers of genes. Can we say that chromosome is the physical inheritance of, it represents the physical basis of inheritance? Yes, because they are the physical carriers of genes. So if this is the gene, if this is the chromosome, right? So this is, let's say this is a gene for tallness, right? And because chromosome occur in pairs, now they will apply, they'll conclude that whatever Mendel said about factors or alleles, the same thing applies to the chromosomes. Mendel said factors occur in pairs. So chromosomes also occur in pairs. Yes. So factors occur in pairs because chromosome occurs in pairs. This is what Sutton and Boveri said, right? Then Mendel said there is law of segregation. During the gamete formation, only one of the factor will be present. Law of purity of gametes. Sutton and Boveri said, why does it happen? Why do these factors segregate? It is because the chromosomes segregate. See here. So during anaphase of meiosis 1, the two chromosome pairs can align at the metaphase state independently to each other, right? So in that case, what will happen? When the chromosomes will segregate, the alleles will automatically segregate. Is that clear? So read this. This table is very important. They occur in pairs, they occur in pairs. Segregate at the time of gamete formation, that only one of each is transmitted to the gamete. And same, only one of the each pair is transmitted to the gamete. Is that clear? Then independent pairs segregate independently. Independent pairs segregate independently. Only one pair segregates independently of the other. Can you tell which of these columns A and B represent the chromosome and which one represents the gene? Now this is for you to answer and give me a logic also. Yes, guys. So basically what Sutton and Boveri said. Yes. So Sutton and Boveri has combined the Mendelian information. Is that clear? Is that clear to all? Now, so see, these are the important lines. So Sutton and Boveri, it was given by Sutton and Boveri that pairing and separation of pair of chromosomes would lead to the segregation of a pair of factors they carry. So important line, united the knowledge of chromosomal segregation with Mendelian principles and called it chromosomal theory of inheritance. And please watch carefully. It is Sutton who united. Now this is the point to be taken care of. Chromosomal theory was given by both, but who united the knowledge, the name is Sutton and not Boveri. These are some small points that you have to remember. Is that clear? Theory was given by both, but who united the knowledge? If somebody asks you, the question appears, it is Sutton. So they can confuse you by writing Boveri and you might think, okay, they were together in all the things. No. Is that clear? Please tell me that these points are clear. Now, Sutton and Boveri have done their work, but it has to be experimentally proven. Somebody else will come up and experiment it. It will confirm the theory, right? So who did that? It was Morgan. So experimental verification of the chromosomal theory was done by Thomas Hunt Morgan and his colleagues. And which organism did they use? It was Drosophila melanogaster. Now the point is, why did they use Drosophila melanogaster only? Now there are some reasons given here. Morgan worked through the tiny fruit fly, that is Drosophila melanogaster, for certain reasons. Number one, it can be grown easily on the simple synthetic medium. Number one. Second, short life cycle which completes within just 2 weeks. Third, single mating will give you large number of progeny. So if the progeny size is large, you can get to see different combinations and that many combinations. Fourth, there is clear differentiation between male and female. There is sexual dimorphism. You can see that. And they are easily distinguishable. And fifth, many types of hereditary variations can be seen with the low power microscope. These are the 5 reasons why Morgan chose Drosophila melanogaster as an experimental organism. Is that point clear to all? Please tell me. Is that clear to all? Now, while Morgan confirmed the chromosomal theory, and during his experiments on Drosophila, many experiments on Drosophila, he also came across another phenomena. Now this phenomena becomes an exceptional case to law of independent assortment. Is that clear? What did law of independent assortment say? Law of independent assortment said that out of the 2 pairs, when we are considering more than one pair of genes, in that case, any allele can go with the allele of the other gene independently. So they align themselves, they assort themselves independent of the other allele. Is that clear? This is what Mendel said. But Morgan found out something else. Now, because the chromosomal theory has been established, it has been established that chromosomes are going to carry genes or alleles, right? So in that case, Morgan found out this concept. So he gave this concept as well as coined the term. So Morgan discovered linkage as well as coined the term linkage. So both the things were done by Morgan. Now, while experimenting, see, he has made these very important observations. So while he studied, see, in cross A, first of all, in cross A, he is considered, look very carefully, he has studied body color and eye color of Drosophila. And in cross B, he has considered eye color with the wing size. Is that clear? So first of all, you must know, see, I'm going to cross these. So understand very carefully because these colorful diagrams get little confusing. So what do you have to study in this? See here. So when he considered body color and eye color, he chose a female which has yellow body color with white eyes and male with wild type. That means brown body and red eyes. This was the wild type actually. Okay, brown body and red eyes. Is that clear? So when he crossed and he did that typical dihybrid cross, he found out that the parental generation is found out to be 98.7. Do you remember in Mendelian dihybrid cross, Mendel's dihybrid cross, we have studied that parental frequency was 62.5% and recombinant's frequency was around 37.5%. Do you remember? You calculated this yourself. 10 out of 16 and 6 out of 16. Do you remember that conclusion? Yes or no? Right? That was Mendel's cross. Now Morgan found out that when he crossed this, when he considered body color and eye color, in that case, the parental frequency is coming out to be 98.7 and recombinant frequency is coming out to be 1.3%. This frequency happens when law of independent assortment applies. When law of independent assortment applies. So in that case, if there are 4 gametes formed, capital A, capital B, capital A, small b, right? Small a, capital B, and small a, small b. We say that they are independently assorting themselves. There are equal chances of everything happening. So all the gametes will be found in equal frequency. All will be formed in the same percentage. Yes, but when he is taking these two characters, he is observing that there is a very strong deviation from the original percentage, which is 62.5 and 37.5 respectively. Here he is getting from 62.5%, the deviation is 98.7, and from 37.5% of recombinants, he is getting 1.3%. Is that clear? But then he did the other cross, in which he is considering eye color with the wing size, male and female. So you must know what type of female has he chosen. Eye color white and miniature wings, small wings. While the male is chosen with red eyes and large wing size. This is the male. Is that clear? So the question comes up, what is the phenotype of female when he was considering this cross? And when he was considering this cross, what is the parental frequency or what is the recombinant frequency? So see here, when he did this cross between eye color and wing size between male and female, taking these genotypes, the parental type comes out to be 62.8, which is almost the same as 62.5. And here he is getting the recombinant frequency of 37.2, which is similar to 37.5. We can take it as same. What do we conclude? This part, K cross A, is not following not obeying the law of independent assortment. Please tell me these points are clear. Not obeying or following the law of independent assortment. While this case, it obeys law of independent assortment. So if it is not obeying the law of independent assortment, then what is it doing? It is following the concept of linkage. This is the case of linkage. What does linkage say? Linkage means that genes are linked together. According to Mendel, there is equal probability of capital A traveling with capital B as well as probability of A going with small b. That is why we are getting both in the equal percentage. Are you getting that? But Morgan will conclude that because, see, why does he conclude that and how does he say that? Because he says that chromosomes are the carriers of genes, right? So now because chromosomes are the carriers of alleles, let's say it is this, or I can take it as A and I'll take it as small a, and I'll take this as B and this is small b. Now, because according to Morgan, like it was according to Sutton and Bovary, and Morgan has confirmed that experimentally. Now, because these A and B are present on the same chromosome and chromosome is going to segregate, so what will happen? There is higher probability of capital A going with capital B. And similarly, there's higher probability of small a going with small b because the chromosome will segregate and not the genes. Do you understand? Please tell me this point. Yes, guys. Are you able to understand what is happening here? Quickly, guys, let me know. Do you understand? Yes or no? Can we proceed now? Is that clear? So this is Mendel, but according to Morgan, capital A will always go with capital B, small a will always go with or prefer to go with small b. There is very less chance that A will go with small. Now, when will? Now, just let me know. Use your brains. What is the probability or why and how with A go with small b? This is only possible when crossing over takes place. Yes or no? Yes or no? Right? So, capital A can go with small b and small a can go with capital B. This is a product of crossing over only. Yes or no? Are we understanding this point? Yes. Now, please understand. Now, came Sturtevant. Now, Sturtevant was a student of Morgan. Sturtevant said that the recombination frequency gives us the distance between the two genes. It is equal to distance between the two genes. Is that clear? What did he say? So, for example, if let's say the two genes are being considered here, let's say here. So, what is the frequency? The recombinant frequency here is, the recombinant frequency here is 37.2, right? While here the recombinant frequency is 1.3. So, this recombinant frequency gives us the distance between the two genes. That means if I make this if I make this chromosome, so the gene for body color and the gene for eye color, this is eye color. Is that clear? So the distance between the two genes, this distance, what is the distance between the two genes? It is 1.3. The unit is cm, centimorgan. That is what he said. Do you understand? And then if I take eye color and wing size, so this is eye color and wing size, let it be wing size here. So what is the distance between eye color and wing size? Equal to the recombinant frequency. So this distance would be 37.2 centimorgan. So this was explained by Sturtevant. Please tell me these points are clear. I am sure, guys, that you have not read linkage like this before. I can claim this. You have not read linkage like this before. Yes or no? Have you ever observed this with this perspective? Please tell me. Have you ever observed these diagrams from this perspective? Yes or no? Is that clear? All right. Let's move on now to the next part. So this is Sturtevant giving us, and the genetic maps, and the distance is measured in unit, measured as cm, that is centimorgan. Is it clear? So you can draw the genetic maps to find out the distance. Now coming on to a very important topic, that is sex determination. Let's see here. So guys, sex determination occurs by or happens by three different phenomena. So first phenomena is environmental factors controlling it. Then we have haplodiploidic method, and third is the sex chromosomes. So for environmental factors, number one, it is the temperature. So there are some species of animals in which higher temperature will give rise to male sex, and then lower temperature can produce females, or the vice versa, right? So temperature can be one environmental factor, and second could be the presence of other gender. So for example, let's say if an egg hatches and larva comes out. Now, if there is already a male present in the vicinity, this larva will develop into a female. Is that clear? But if there is a female in the vicinity, then this larva will change into a male. So this is how they choose their, or they determine their sex. Is that clear? Then second, this is actually not to be discussed in detail, not given in your NCERT. Second method of sex determination, which is observed in case of honeybees, or apis, the scientific name for honeybee. This is haplodiploidic method. So in that case, basically, the male is haploid and the female is diploid. Is that clear? So female is diploid. In case of honeybees, guys, so honeybee, 2n is equal to 32, and in males are drones, n is equal to 16. Is that clear? So how does it happen? So basically there are, let's say in honeybee, what happens? So queen lays the eggs, right? So queen lays the eggs. Now see, queen is a female, so she is 2n is equal to 32. Is that clear? So if she is going to produce eggs, eggs are of course haploid because they are produced as a result of meiosis, right? So eggs are produced haploid because they are formed as a result of meiosis. Now we have a male here. So male is known as a drone. So male is the drone, and drone is haploid, n is equal to 16. Now if a haploid produces sperms, so from haploid to haploid, sperms are haploid of course, what will be the process involved? It will be mitosis. So first question here, with what process does drone produces the sperms, and which cell division is involved in case of egg production? You should be clear about it. Yes. Now this one is clear. Now see what happens. There are two probabilities. One, that some of the eggs can get fertilized with the sperms. So here normal fertilization, syngamy, occurs. So when fertilization occurs, it will result in the formation of a 2n structure, and 2n structure will always be female. This can either be queen or it can be worker. So both queen and workers are produced by typical sexual reproduction. This is a case of typical sexual reproduction. Yes, case of sexual reproduction. Am I clear? Now, there is another probability that these eggs might not get fertilized. If there is no fertilization, guys, in case of no fertilization, in case of no fertilization, what will happen? Then only egg will develop into, egg develops without fertilization, that means develops by parthenogenesis. So N will give rise to N. So it is N will produce N organism, which is going to be a male. So how is male produced? Male is always produced as a result of parthenogenesis. Are we clear till here? So it is the type of reproduction that will determine whether it is a male or the female getting produced. Are we clear? Right? And now we have third method, which is by the help of sex chromosomes. So we must know that in all of us there are two types of chromosomes present. So chromosomes can be autosomes, which are not involved in sex determination, and the typical chromosome which is involved in sex determination is called allosome or sex chromosome. So if we talk about humans, we have 22 pairs, yani 44 chromosomes which are autosomes, and we have one pair, that is 2 chromosomes which are involved in sex determination, hence we call them sex chromosomes. So there are two types, X and Y. So depending upon which combination occurs, if it is XX, then it is a female. In case of humans, I'm talking about, if it is XY, that will make a male child. That is only in case of humans. But here we are going to talk about some other organisms as well, in which sex chromosomes are involved in the determination of the gender. Now see here. So we have first type, XXXY type. Now this type is seen, see, XX is the female and XY is the male, we have just seen. This case is seen in Drosophila and humans. Is that clear? So see, this is the male, this is the female. So when the female will produce gametes, she is going to produce same type of gametes. Both eggs will contain XX only. During law of segregation, gametes will carry only one. But when male is going to produce sperms, though there will be some sperms, 50% sperms will carry X chromosome, while other 50 will carry Y chromosome. So male is producing two different types of sperms. Yes or no? Right? So male is called heterogametic. So here the male is heterogametic because producing two types of gametes, while the female is called homogametic. Yes or no? Do we understand this? Yes. In other case, now see. So basically, now let's complete this point. So basically, it is the sperm. It depends upon which sperm is going to fertilize the egg. If X, so this is the X containing sperm, this is the Y containing sperm, and here we have only one type of egg. So it depends which sperm will fertilize the egg. So the underlining statement is, it is always the heterogametic partner. It is always the heterogametic partner that is responsible. Sex chromosomes heterogametic partner is responsible for the sex, determines the sex. Are these points clear? Is this point clear to all? It is always the heterogametic. So guys, in this case then, who is responsible? It is the male. That determines the sex. Now coming on to another type, we have XXXO type. Here again, this is the female and this is the male. So female is again homogametic and male is again heterogametic. This case is seen in case of few insects. See, Drosophila is also an insect, but it has human-like sex determination method. Few insects like grasshopper, cockroach, they show this phenomena. Now, what is XO? XO means nothing. So, here the male is going to produce two types of sperms. One containing X chromosome, other containing nothing. There will be no X chromosome. 0 means no X chromosome. Is that clear? So again, which sperm will hit the egg, that will determine. So again, here also it is the male that is going to determine, because male is the heterogametic partner here. Now, in case of birds, guys, birds or fowls, one and the same thing, or chicks are birds, of course, right? We have a different mechanism, ZWZZ type. So here, female is ZW and male has ZZ type. So, who is homogametic here? Male is homogametic. And who is heterogametic? Female is heterogametic. So, guys, in birds, it is the female that is responsible for sex determination. Yes or no? Then in case of ZZ and ZY type, ZO type, again, this is the female, this is the male. So male will have 50% sperms carrying Z and 50% not carrying Z. Now this is not given in NCERT, but this is seen in case of moths and butterflies. So male is again heterogametic. So you just have to remember one case, and that is of birds, guys, where female is responsible for sex determination, while in rest all the types, it is usually the male who is heterogametic partner and hence determining the sex. Please tell me that this point is clear to all. Yes, guys. Is that point clear? And before I proceed, I again want to tell you that you have to attend all the special classes now, and you have to take all the All India mock test series, right? Special classes time will be 2 onwards again, except on the days when there is AIMT, and we have to practice all type of questions before we take the Re-NEET exam. Is that clear? So put an alarm for 2 p.m. where we will be conducting special classes, practicing the questions. So follow me on Unacademy Learners app and you can solve questions with me. Is that clear? All right. Moving on to the next phenomena, very important. This is pedigree analysis. Now watch carefully. Again, see guys. So the first genetic tool that we have studied is that of test cross, where we are able to determine the genotype of the dominant trait. Now, in pedigree analysis, guys, we can study the presence of trait, study the inheritance of trait through a family, in the whole family, or in the future generation. Is that clear? So if you want to study the inheritance of trait in the whole family and whether it will be present in the coming generation and what is the percentage, what is the probability of it occurring in the future generation, we have this very important genetic tool. It is again a good genetic tool. So we make a family tree. We make a family tree here. It is a genetic tool. Is that clear? So we have made some symbols. We use some symbols. Square is for male, circle is for female, and when we put it this way, that is a rhomboid form, that means we are not sure whether the child is male or female. So the child was born, whether it was male or female, we don't know about, that is sex unspecified. If we shade that square or circle or this rhomboid, this means the individual is showing the presence of that trait. Affected means it is not always a disease, it can be anything. We can be talking about any particular trait. So it shows the presence. 1 line means mating, 2 lines means mating between relatives or consanguineous mating. That means mating between cousins, relatives. Is that clear? Then this was a PYQ. So you should be able to identify the symbols, guys. This has already come up in the exam. Then if we draw this symbol, that means parent, male, father, mother, and they are having progeny. So the order in which this circle and square is drawn, it means the order of the progeny. See, it is in the order of birth, left to right. And then parents with just 1 child and the male child is affected here. This is how they show. And if they are not sure about the sex of the children of the progeny, but they know that 5 children were produced, so when the sex is unspecified, how do we put it? This. And we put the number of progeny within that box. Is that clear? Please tell me these symbols are clear to you all. Yes? Now, before we learn how to use this, guys, we have to understand there are different types of genetic disorders which can be studied by the help of pedigree analysis. So first, before going into the details, let's understand the type of genetic disorders. One is Mendelian disorders and second is the chromosomal disorders. What is the difference? So Mendelian disorder is generally caused by the alteration in a single pair. For example, we have sickle cell anemia, right? GAG to GUG. So just single base pair, single gene alteration leads to Mendelian disorders, right? While if there is whole chromosome involved, that means say there is an extra chromosome available present, or while we should have 2 copies of every chromosome because we are diploid, so instead of having 2 we have 4 or we have 3, or we don't have any copy of that particular chromosome, both the copies are absent, or 1 copy is absent. Is it clear? That is called chromosomal disorder. So chromosomal disorder again of 2 types, aneuploidy due to the non-disjunction, means failure of segregation of a particular chromosome, and if it is a failure of segregation of the complete set, so we are 2n. So instead of 2n, what happens? Let's say we become 3n or we become 4n, right? So what will happen? That is called polyploidy. Now polyploidy is not seen in humans, because if an embryo has if embryo shows polyploidy, that embryo doesn't sustain. But we do get to see this polyploidy situation in plants. Is that clear? So in humans, in animals also it is formed, but it is not sustained. Nature has put an end to that. And then aneuploidy is definitely seen, and we'll study the cases. So here, see, 2n plus 1 means normally there are 2n chromosomes, but then there is an extra 1 chromosome, then extra 2 chromosomes, then 1 less chromosome, 2 less chromosomes. Are you getting that? So this is what aneuploidy is. Is that clear to all? Please tell me. And then in Mendelian genetics, there is alteration of single gene, guys. These can be further of 4 types. Please understand. Autosomal dominant. So autosomal dominant means the single gene which is altered is present on the autosomes. 22 pairs of chromosomes in humans, for example. And it occurs in the dominant state. So if the gene is in the dominant state, the trait will occur. For example, myotonic dystrophy. These examples are very important. Then autosomal recessive, sickle cell anemia, thalassemia, PKU, all these are autosomal recessive. And then we have sex-linked dominant, for example, hypertrichosis. Now sex-linked, sex means these genes will be present on X and Y. So this one that I have discussed, not really in your syllabus, but this one is on Y chromosome, and Y chromosome is only in males, so this will only be seen in case of males. So what is hypertrichosis? You will see excessive hair growth on ears. Is that clear? Then sex-linked recessive. Again, this can be X-linked also, Y-linked also. I am just discussing X-linked recessive. So X-linked recessive is color blindness and hemophilia. Is that clear? So now let's talk about, see, I have discussed these few of these diseases. I have talked about sickle cell anemia. First of all of them are not in your syllabus. So we are only going to talk about myotonic dystrophy, sickle cell anemia already discussed, PKU already discussed. I will talk about thalassemia, and then I'll talk about these. Is this clear? Now, let's first discuss, see, sickle cell anemia is done, PKU is done. In myotonic dystrophy, see, it's a type of muscular dystrophy. So there is muscle degeneration, muscular dystrophy. Is that clear? And it is a genetic disorder. It occurs in a dominant state. Let's talk about thalassemia here. So thalassemia. Remember, we have two types of chains in hemoglobin, 2 alpha and 2 beta. So alpha chain and beta chain. Is that clear? So alpha chain is produced by, guys, this chain is produced by 2 genes. One is HBA1 and the second is HBA2. There are 2 genes involved in forming the alpha chain. And these 2 genes are present on chromosome number 16. Is that clear? Beta chain is formed because of just 1 gene, and that gene is HBB. Is this clear? And this 1 gene is found on or present on on chromosome number 11. So I always had this confusion to remember which two genes are on chromosome 16 and 11. So I use a very naive trick, which is that it is a bigger number, so it will carry two genes. There is no logic here. It is just a method to remember. So is that present two genes on chromosome 16 and one HBB present on chromosome number 11 here. Now what is thalassemia? So guys, thalassemia can be of two types, thalassemia A and B. So in A type, alpha chain is not formed, and in B type, beta chain is not formed. And then there is C type. In C type, both alpha and beta are not formed. Why are these chains not formed? Of course, because if there is any change, any mutation in these genes, alpha will not be formed. If there is any mutation in these genes, beta will not be formed. And if both are affected, both will not be formed. Is that clear? Now, here in thalassemia, compared to sickle cell anemia, where the chemical composition of beta chain is changing, right? So there the quality of beta chain will be different, while here the beta chain itself will not be formed. So quantity is different. So that is why NCERT is writing this line that sickle cell anemia is more of a qualitative disorder. Here the quality is different from GAG to GO, glutamic acid to valine. So beta chain, the nature of beta chain is destroyed, but it is formed. But in case of this thalassemia, it is more of a quantitative disorder where the whole chain is not formed. Quantitative disorder. Please tell me that you are clear about these points. Yes guys, please tell me these points are clear to all. Are we clear? So once again, so thalassemia, it is an autosomal recessive disorder. Why autosomal? Because we see the chromosomes 16 and 11 are involved. Here it is a recessive disorder. It will only occur when it is in a homozygous recessive state. Then there are three types, A, B and C. In A type, alpha chain is not formed, B type, beta chain is not formed, and C type, both are not formed. See, alpha chain normally is formed by two genes, HBA1, HBA2. These two genes are present on chromosome number 16. Beta chain is formed by one gene, HBB, present on chromosome number 11. Is that clear? Any mutation, any alteration will give rise to thalassemia. So, very important to remember, in sickle cell anemia, it is a qualitative disorder. Beta chain is formed, but the beta chain is not normal. Sixth amino acid is changed, glutamic acid to valine, but it is formed. But in thalassemia, there is a possibility that beta chain may not be formed, or alpha may not be formed, or both are not formed. So it is more of a quantitative disorder. Very, very important point. So please let me know that these points are clear to you all. Is that clear? Is that clear to all? Now moving on to pedigree analysis once again. So guys, how do we analyze? So see, it is first of all very clearly mentioned here. Wait, this has been mentioned clearly that this one, see, it represents autosomal dominant trait, which is myotonic dystrophy. This is a case of this, and this represents sickle cell anemia, which is an autosomal recessive trait. But if you get a random pedigree, just forget about this for a while that it is written. So if you get a very random pedigree, how to determine whether it is autosomal or it is sex-linked, and also how to determine whether it is a dominant trait or a recessive trait. So how to do that? The point is this. So step number one, guys, step number one is you have to see whether it is recessive or it is dominant. Now, in order to determine whether it is recessive disorder or a dominant disorder, see all the generations. It will take me time. 1.5 hour. Please eat and sleep. Now, the first step is recessive dominant. So, when you have to determine whether this trait is dominant or recessive, so how do we determine that? Now, the first step is see all the generations. See all the generations. See here. Number 1. This is first generation. This is second generation. This is third generation. Or let's say this is the third generation. Okay. This is the third generation. First, second and third generation. Is that clear? Now, the red ones means they are affected. Okay. That means they show a presence of a trait. Now, how do we determine whether it is a recessive disorder or a dominant disorder? So, you have to check for all the generations. If that trait is present in all the generations, guys, if the trait is present in the parent and present in the next generation also, that means it is a dominant trait. If parents have it and the children don't have it, or parents don't have it and children have it, so any one generation is skipping that trait, it is 100% recessive. If the generation is skipped, if a generation is skipped in the inheritance of trait, that is a recessive trait. Is that clear? Now, for example, here, see here. So, generation 1, one of the parent is affected. Now, see their children. This girl child is unaffected, male child is affected, male is unaffected, and the girl child is affected. So, parents have it, some of the children have it. Not necessarily all of them have it, but some of the children show it. Now, see in the third generation. Now, this girl marries an unaffected male and one of the child is affected. Yes. So, this generation has it, this generation has it. Talking about this one. So, both the parents are unaffected. So, none of their children is also affected. Yes or no? So, do we see that this is a case of dominance? Here, no generation is skipped. If parents have it, the children have it. Is that clear? So, this is a case of dominant trait. Keep it aside whether it is autosomal or recessive, but this is fixed that this one is a dominant trait, whatever it be. Coming on to this one here. Now, this is generation 1, this is generation 2. Now, using the same formula, parents do not show that trait, but the trait appears in 2 of their children. Now, see here, this third child, the girl, marries an unaffected male, but again, the trait appears in one of the children. So, the generation is being skipped, not in parents, expressed in children, not in parents, expressed in children. Whatever the trait is, it is skipping a generation, hence it is recessive. Please tell me these points are clear to you. So, this is the trick to determine whether it is dominant or recessive. Clear to all? Then, guys, moving on to the second step. So, second step, we are going to determine whether it is autosomal or whether it is sex-linked. Is that clear? Now, how do we determine autosomal and sex-linked? See, in autosomal, guys, there is no fixed pattern. No fixed pattern. That means mother to daughter or male child. It can be either way. Father is passing on the trait to girl also, to the boy also in random frequency. So there is no fixed pattern followed. But in case of sex linked, guys, there can be 2 situations. If it is Y linked, that will always be passed on to the all boys, because man is always going to pass on that Y chromosome to his son. So if Y has that gene, it will be passed on to the boy. Yes or no? So if it is Y linked, it will always be present in all boys. Is that clear? But if it is X linked, then X linked inheritance shows a particular inheritance which is called criss-cross inheritance. So in criss-cross inheritance, what happens? In criss-cross inheritance, see what happens. Here, the mother passes on the trait to her son, and son gives it to his daughter. This is criss-cross. And similarly, father gives that trait to the daughter and daughter gives it to her son. Please tell me these points are clear. So now in both these pedigrees, do you see any fixed pattern? So see here, mother is affected, giving the disease to male as well as female. Female is affected, she is giving the disease to the female. But here there is no pattern, see, no crisscross. Otherwise here, if the mother had the trait, she should have given it only to the son and not to the daughter, right? Or the daughter could have been a carrier, but definitely not affected. And here, mother giving it only to the daughter, not giving it to the son. So 100% this is autosomal and not sex-linked. Similarly here, not in either of the parents, but it is occurring in male and female at the equal frequency. And here, neither have the disease and it is suddenly occurring in the male. Do we see that? So again, this is also autosomal recessive. Please tell me these points are clear to you all. If you want to practice more such questions, guys, with me, don't forget to join me in special classes. We are going to practice lots of pedigrees. Lots of pedigrees. Is that clear? I can make sure that at least this question, like other questions also, your pedigree question is also not going wrong at all. Whatever the question is, we can make that sure by more practice. Is that clear? So guys, other than this, so I have to talk about, see, color blindness. Let me talk about color blindness, which is a sex-linked recessive disorder. Now see, how do these occur? And I've told you sex-linked recessive disorders, they always follow the pattern of crisscross inheritance. Is it clear? So there are two examples. One is color blindness, where the person fails to distinguish between red, green or blue color, that is color blindness. And then there is hemophilia. This is a bleeding disorder where a minor bleeding can turn fatal also, or very serious, because the clotting factors are not present in here. So talking about color blindness. So this gene for color blindness is present only on X chromosome. So it is X related, X linked. This is X linked. Is that clear? Now, the gene for color blindness is present only on X. So we have three conditions for females. So if it is in this homozygous recessive state, such female is color blind female. If she has this only on one chromosome, X chromosome, she doesn't have a disease, but she is a carrier because she can pass it on to the next generation, but not diseased. She has a normal vision. While this is absolutely normal female, like you and me, most of it. And then in case of males, guys, this one is a normal male, right? And if male has it even on one, because male has only one X chromosome, so if even one X chromosome carries the gene for color blindness, male will be affected. So this will be a color blind male. Is that clear? So in males and females, we have different ways. So this female is called heterozygous or carrier. But in case of males, guys, we don't call it heterozygous, we call it hemizygous. So hemizygous is the affected male. Is that clear? So now can you cross which male and female, if crossed, can produce type of progeny? So what is the probability if this crosses with this or this crosses with this? So you can solve such questions. And again, we'll be solving all those questions in the special classes. Then coming on to hemophilia. Again, in hemophilia, we have three situations in case of females: XH, XH, XHX and XX. So this is the normal female. This one is heterozygous, so she is a carrier, definitely not diseased. And this one is a hemophilic female, usually doesn't survive long. Is that clear? And then we have XY and XHY. So this is the normal male. But if even one of the X chromosomes, male has only one X chromosome, so if it is present in the hemizygous state, male will be hemophilic. Now, can you answer why the population has more males affected with sex-linked, X-linked disorders? Why? Because see, this homozygous situation is always a rare condition. So if even even if male has only this hemizygous situation, so male can be easily affected, while the female in a similar situation will only be carrier and not the diseased. Is that clear? So males are always more affected with such situations. Please tell me that you understand this point very clearly. Yes or no? Do we understand? So males are always more affected in this case. And now guys, coming on to chromosomal disorders. So we have 3 chromosomal disorders to be talked about. This is Down syndrome, studied by Langdon Down syndrome. So what happens in Down syndrome basically? So the 21st chromosome, which is normally in just 2 copies, in such individuals, this occurs in 3 copies. So there is an additional copy of 21st chromosome. We call it trisomy of 21st chromosome. So what are the repercussions of these? So the repercussions are such Down syndrome has flat back of the head, many loops on the fingertips, characteristic palm crease, congenital heart disease, big and wrinkled tongue, and broad flat palm. So when there is trisomy, that means one additional copy of 21st chromosome, so the total number of chromosomes would be not 46 but 47. So this shows the karyotype, and do you see this is the 21st chromosome which is found in 3 copies here. Is that clear? Then talking about this Klinefelter's syndrome. See, Klinefelter's syndrome, and this one is the Turner's syndrome. This one is the Turner's syndrome. So what happens in Klinefelter's and in Turner syndrome? See, in Klinefelter syndrome, the condition is XXY. So it is the trisomy of sex chromosome. Yes, trisomy of X chromosome. So total chromosomes here again is 47, right? While this is Turner syndrome. See, the names are after the name of the scientists who have studied them. Turner syndrome is because of XO condition. While the normal female is XX, Turner's female is XO. She just has one X chromosome. So there is a monosomy. Monosomy means single copy. Monosomy of X chromosome seen here. So what will be the total number of chromosomes? Instead of 46, she'll have one less chromosome. Is this point clear? So what are the other features? So in Klinefelter's syndrome, guys, the male will have a normal masculine structure, tall, but few feminized characteristics like development of breast, which is called gynecomastia. Development of breast is called gynecomastia, and testes also non-functional, rudimentary, so sterile. Is that clear? Infertile. Like in case of Turner syndrome, she has short stature, overall feminine, but of course underdeveloped feminine characteristics, masculine characteristics. He'll develop feminine characteristics, she'll develop masculine characters, and ovaries again non-functional, rudimentary means non-functional, hence she is also sterile, means non-fertile. Are these points clear? So guys, with this, the first chapter is done. First chapter of genetics is done. Quickly let me know that these points are clear to you all. And once again, I'm reminding you guys that this Sunday, 7th June, we have to take All India mock test for real NEET experience, performance insights, and to improve your score in the Re-NEET exam. Is that clear? Now, moving on to the next chapter, which is molecular basis of inheritance. Now, this chapter I have made all in the form of flowcharts. So, this will be easy for us to quickly revise. Now, see, this is molecular basis. In previous chapter, while we were studying chromosomal theory of inheritance, we studied that chromosomes are the physical basis of inheritance. Yes. This is the molecular basis. Which molecule is responsible, or you can say the chemical basis is DNA, right? Now see, let's talk about DNA. DNA, deoxyribonucleic acid. It is the largest biomolecule out of proteins, fats, carbohydrates. DNA is the largest biomolecule. Earlier it was known as nuclein and later known as nucleic acid. Is that clear? The term was given by Frederick Mischer because it is found in nucleus, and acidic, it was called a nucleic acid because of the acidic nature. Is that clear? Now, DNA will act as a genetic material. So see here, what is DNA made up of? It is made up of deoxyribonucleotides. Is that clear? So it is a polymer of these. So basically, deoxyribonucleotide is a monomer of DNA. So you combine these, you make a polynucleotide chain. Now, number of nucleotides will be fixed for each species. So how many nucleotides are present in you or in E. coli or in this, that is fixed, and you have to remember these. Okay. Now, each nucleotide, guys, is made up of 3 components. We have one nitrogenous base, which can be ATGC, adenine, thymine, guanine, cytosine, and a pentose sugar, which is deoxyribose, and phosphoric acid. Nitrogenous bases can be purines and pyrimidines. Purines are A and G, and this is T and C. Is that clear? These are pyrimides. Now, so you have to remember that this DNA can be found either as single-stranded DNA or double-stranded DNA. So, single-stranded DNA is observed in 5174 bacteriophage, while double-stranded is seen in lambda bacteriophage, E. coli, and humans. Now, what is the number of nucleotides in all of these? This is your homework to be found out. Please do this homework, and if you can't learn these names, let me know in the comment section. I'll make a special video for that. I have a trick how to remember, but you have to tell me in the comment section for that. Is that clear? Now, structure of DNA. Coming on to, see, it is a polynucleotide chain. It is a polynucleotide chain. That means many nucleotides are going to club together. So what is a nucleotide made up of? 3 components, sugar in between, and phosphoric acid and nitrogenous base. So look here, this is sugar, pentose sugar. Do we see 1, 2, 3, 4 and 5 carbons here? And now to carbon number 1 is attached the nitrogenous base. The bond formed is glycosidic bond, and to 5th carbon is the phosphoric acid attached. Now this bond is known as phosphoester bond. So this is one nucleotide. This is one nucleotide. Are we clear? Similarly, we have another nucleotide here, right? We have another nucleotide. So one nucleotide, another nucleotide, they are connected through carbon number 3. And now the bond that connects the 2 nucleotides, that is a phosphodiester bond. And guys, remember, if you have to calculate the number of phosphoester bonds, you will include phosphodiester there. If you have to calculate the number of phosphoester bonds, is that clear? You have to count the phosphodiester bond and equal it to 2 phosphoester bonds. Now, for example, if the question says that there are 3 nucleotides connected to each other, is that clear? Or 3 nucleotides bonded, calculate the number of phosphoester bonds. Can you do that? So what do? We calculate this phosphoester bond, another phosphoester bond, and in 3 nucleotides, there will be 2 phosphodiester bonds. So 1, 2, 3, 4, 5, 6. Do we understand this? Please tell me, yes or no. So this is the phosphodiester bond. Clear to everyone? Yes, please. Now, if we have to break the DNA chain, if we have to separate these nucleotides, guys, so we have to break phosphodiester bond. Now, this bond can be broken by the restriction enzymes, and this is a very important point. Tomorrow when we study biotechnology, we are going to use these enzymes. And if we want to combine the nucleotides, we want to join the DNA fragments, we want to join the nucleotides, the enzyme which is used for joining these is DNA ligase. So remember, which bond will be broken by restriction enzyme and which enzyme is going to form that bond as well. Is that clear? Now, moving on to the next. So, see, this is the question. So, you can get such questions. Calculate the number of following bonds in a trinucleotide. Glycosidic bonds are 3. And then phosphoester bonds, I told you, 7. Phosphodiester will be 2. And H bonds will be how many? So, you have to calculate in such a way. Is that point clear? Right? So we know that this is one polynucleotide chain, and now see, usually the DNA is double-stranded. So when the two strands of DNA are connecting, the 2 strands are always anti-parallel. Now see here, why it becomes anti-parallel? The anti-parallel nature is actually because of the sugar. So do you see, in one chain, this polarity, do you see the polarity of sugar? And now here, do you see the polarity of sugar? Yes. So it is because of the opposite polarity of sugar that the two DNA strands are anti-parallel and opposite to each other, anti to each other. Parallel nature will be decided in a while. Now see, one side is called the 5' end, which side? The one which has phosphoric acid, free phosphoric acid, and the other side which has free 3' end, 3, and it is OH group. 3OH group is free. We call that side as 3A' end. Is that clear? Now, on the other side, we'll have 3OH group free on this side. So this is the 3' end, and here we have phosphoric acid free on the 5' end. This will be the 5' end. Is that clear? Now, how are the two strands connected? They are connected through their nitrogenous bases. The two nitrogenous bases will be joined by hydrogen bonds. For example, it is always one purine getting connected with pyrimidine. So purine and pyrimidine connected through hydrogen bonds. Is that clear? Now, why they are parallel to each other? Because purine always binds with pyrimidine. Purine has 9 members. This has 6 members. So one side we have a larger entity, that is purine. On the opposite side, opposite to that, we have a smaller one, pyrimidine. If this side we have pyrimidine, then opposite to it will be purine. So one big, one small, one small, one big. So it will always maintain the same distance between the two strands. So that will give it a parallel nature. So the two strands are parallel because of purine-pyrimidine combination, one big, one small, and anti because of the opposite polarity of sugar. Is that clear? Now, see, if A binds with T, this is purine again, this is pyrimidine again. Purine, this adenine binds with thymine with 2 hydrogen bonds, and guanine bonds with cytosine with 3 hydrogen bonds. So which one is stronger? This is of course more stable. So if DNA has more GC content, that DNA will obviously be more stable. Now, why I wanted to discuss genetics before biotechnology? You tell me, if I have to separate the two DNA strands, which bond is getting broken? Quickly let me know. If I have to separate the two DNA strands, which bond is getting broken? Yes, Nancy, you are visible. Absolutely visible. But I am focusing on molecular basis of inheritance. Is that clear, guys? Okay. So if we have to separate the two DNA strands, which bond is broken? It is the hydrogen bond. How do we break that? There are multiple ways, but a very basic way of separating them, especially artificial way, that is during PCR, we are going to heat it up. We are going to heat and these hydrogen bonds will be broken. Are you getting that? So that is why we need to understand genetics very carefully and more clearly before biotechnology. So revise it once again, watch it again before tomorrow's class at a quicker speed, at a faster speed. Now see, according to Watson and Crick model, guys, what is the structure of DNA? So first of all, this structure of DNA given by Watson and Crick in 1953 is based on X-ray diffraction method. And now this X-ray diffraction was done by Maurice Wilkins and Rosalind Franklin, and both of them were physicists, right? And this Watson and Crick model gives us a secondary structure of DNA. Secondary means two-dimensional structure of DNA. So according to this Watson and Crick model of DNA, sugar and phosphate forms the backbone of DNA. Nitrogenous bases are placed on the inner side. Most common form will be bDNA. Is that clear? Now, see, I've already written secondary structure. So DNA is a double-stranded, and the two strands are joined by hydrogen bonds. They are anti-parallel due to the opposite polarity of sugar. And why are they parallel? It is parallel due to the purine and pyrimidine combination. Is that clear? It is parallel due to purine-pyrimidine combination. It is anti due to opposite polarity of sugar. It is helical in nature. This is the structure of DNA. Is that clear? So what does this mean? Sugar and phosphate backbone. So sugar, phosphate, sugar, phosphate, sugar, phosphate. So this is the backbone. Nitrogenous base is on the inner side. This is the nitrogenous base. the two strands will be connected through the nitrogenous bases. Do we understand? So while it is going in the helical form, this one is the minor groove, this is the major groove. Now, most common type of DNA, there are 5 types of DNA. The most common type is bDNA. It is a right-handed helix. So the jalebi goes in clockwise direction, right? So the now see the numerical values attached to bDNA. The pitch is 1 complete revolution. The length of one pitch is 3.4 nanometer. Is that clear? And in one complete pitch, guys, there are 10 base pairs. So this, let's say it is A, it is G, this is G, this is C. So AT is one base pair. How many base pairs are present in this one length? That is 3.4 nanometer, 10 base pairs. Now, if I say this total length is 3.4 nanometer, there are 10 base pairs in this length. What is the distance between the two base pairs? Can you calculate? Can you calculate this distance? This is simple maths. It is 3.4 upon 10. The distance between the two would be 0.34 nanometer. Can we say that? And this is known as rise per base pair. Please tell me these points are clear to all. Please tell me these points are clear to all. Yes or no? Is that clear? Now, moving ahead, guys. Moving ahead. So, purines and pyrimidines. See, we know that adenine and guanine are found in both RNA and DNA, and so does the cytosine. Uracil is only in RNA, thymine is only found in DNA. What are the other differences? Purine forms a double ring, like a 9-membered structure. Pyrimidine is a single ring and 6-membered structure, right? There are now Chargaff's rules. So basically, Watson and Crick were able to deduce the structure not only on the basis of the X-ray diffraction. That was obviously one very important tool, and the tool which led them to conclude the structure. But then there were certain other apprehensions before that X-ray diffraction, and they were based on Chargaff's rules. So Chargaff is the name of the scientist, and he gave us certain facts about the DNA, that total amount of purine is equal to amount of pyrimidine. Is that clear? Because we know purine will bind with pyrimidine only. So the amount of purine will be equal to amount of pyrimidine. And we know that A will always bind with T and G will always bind with C. So amount of A will be equal to amount of T. Amount of C will be equal to amount of G. Is that clear? So can I write that total purine, that means A plus G, is equal to C plus T? We can do that. If I do some mathematical manipulation here, so if you bring this factor below as a denominator across this equal to sign, it will go in the denominator. So purine plus upon pyrimidine is equal to A plus G upon C plus T is equal to 1. Can we say that? Please note down. So purine and pyrimidine equal to 1 means they are equal in amount. Yes, but here this is a different equation. Watch very, very carefully. Here I am saying AT upon GC, that how many AT bonds and how many GC bonds are present. AT and GC content, how much AT content and how much GC content will be present in a DNA. This is not 1. This is constant for a species. So compare the two and remember them very carefully. AT upon GC is constant, not 1, while AG upon CT is 1. That means equal. So purine is equal to pyrimidine. Here they are not talking about purine and pyrimidine. So this is constant and this is equal to 1. Is that clear to all? So compare these two equations. Do not make a mistake which is 1 and which is equal to constant. So this is how also you can determine whether the two DNAs belong to the same species or to a different species. Is that clear? Now moving on to, so this is a very basic numerical. If you are given the amount of guanine and you have been asked to calculate the amount of different other nitrogenous bases, can you do that? Now you have been given amount of T is 20. So if T is 20, A is also 20. So A and T, AT becomes 40. The remaining GC, that becomes 60, and G is equal to C. So G is equal to C is equal to 30%. Can we do such questions? Yes, very easy. Now guys, I have discussed this slide in the chapter biomolecules also. See, nitrogenous base, when binds with sugar, either deoxyribose or ribose, depending upon which nitrogenous base it is. Like for example, thiamine will only bind with deoxyribose, uracil will only bind with ribose, while these can bind with both, either. They too, nitrogenous base and sugar, they always form a glycosidic bond, and together they form a nucleoside. So nitrogenous base with sugar, S for sugar, it forms nucleoside. But when we add phosphoric acid to that, when there are three entities, so T for 3, T for nucleotide, and tides are the monomers of DNA. So see their names, you should be able to differentiate that which particular set of names is the name of a nitrogenous base or nucleoside or nucleotide. Is that clear? And then if you add two more phosphoric acids, phosphates to nucleotides, you form the energy sources. Please tell me that these points are clear. So this slide is also very, very important. Now guys, we know that DNA, if we have to calculate the total size of the DNA, see, so you should know how many total nucleotides are present, and I've given you as a homework, you have to learn them, or you have to leave me a comment trick to remember how trick to remember the number of bases or base pairs in every species. There are 4 species that we have to remember. Now we know that according to Watson and Crick model, the distance between the two base pairs, or the rise per base pair, is 0.34 nanometer. Yes. So if you know the number of nucleotides or the base pairs, guys, can you calculate the total length of DNA? Right? Now see, for the haploid content, so this is the haploid number of, this is the number of nucleotides in haploid content of DNA. So that is 3.3 into 10 raised to the 9 base pairs, right? And this is the rise per base pair. If I multiply, that gives me the total length of haploid DNA, 1.1. And what will be the length of the diploid DNA? That will be 2.2 meter, almost the size of dupatta. Imagine this much DNA is present in one cell. And the size of the cell is microscopic. You can't see cell with the naked eye, but the size of DNA is that large. And this DNA is present in the nucleus, the size is even smaller. So 10 raised to the minus 6, right? Size of the nucleus. So how is that possible that such a large molecule is present in such a microscopic structure? So it means for sure that is not extended as it is. It has to be packed and it has to be coiled properly so that it can be packed in such a smaller space, right? So for that, guys, DNA, and also to stabilize it, we know DNA is acidic, histones are basic proteins. Now these histones are going to form an octamer, means 8 molecules of histones will combine together to form an octamer, and these are basic because they are rich in basic amino acids. Very important question and has been repeated multiple times. This is a PYQ. Which amino acids are present in higher amount in histones? Lysine and arginine. So the nature of amino acids will of course determine the nature of the proteins. Is that clear? So histones are lysine and arginine rich. Now these histones are a characteristic proteins of eukaryotes only. Prokaryotes don't have this. So what is found in prokaryotes to stabilize the DNA? We'll talk about that. Is that clear? Now which histones? So see, there are 5 different types of histones: H1, H2A, H2B, H3 and H4. Now these 2A, 2B, 3 and 4, 2 molecules of each will form an octamer. Is that clear? 2 molecules of each will form an octamer, while H1 is going to stabilize the linker DNA. Now see here, this circle which represents H2A, H2A, H2B, H2B, H3, H3, H4, H4, 8 molecules of histones. This structure forms, see here, so this forms the octamer. This structure forms the octamer. Is that clear to what this structure forms the octamer? And on top of that, guys, on this octamer, we have this thread of DNA running around. So DNA thread runs on like this. Do we see? So DNA is acidic. It is negatively charged. Remember that it is negatively charged. and basic, these are positive in nature, positively charged, and together they will form a stable entity. So 8 histones and DNA together forms nucleosome. Please tell me these points are clear to all of you. Yes, guys. Please tell me these points are clear to all of you. Yes, please. Are we clear? Quickly, guys, let me know. Are we clear? Anyone with any doubt till now? Clear? All right. And now in one nucleosome, guys, when this DNA is running around the nucleosome, around the histone octamer, then how many base pairs? See, 1, 2, and how many such base pairs are present? 200 base pairs are present. Is that clear? So nucleosome is called the repeating unit. So this is one nucleosome. Is that clear? So likewise, guys, we have different nucleosomes, and this is the DNA running on that. And now the region of DNA between the two, the region of DNA between the two, this two nucleosomes, this one is called the linker DNA. This is called the linker DNA, which connects the two nucleosomes. Now this is also supposed to be stabilized. So linker DNA is stabilized by histone H1. Is that clear? Now see. So these are nucleosomes, right? Now nucleosomes are going to give it bead-like appearance. So this is a string of linker DNA with nucleosomes occurring as beads on that. It gives it beads on the string appearance, and it forms a chromatin. So this whole thread-like structure is called a chromatin. Is that clear? Now when this thread condenses further, and it condenses further to form a shorter structure called chromatin fibers. And chromatin fibers will condense further to form a rod-like compact structure known as chromosome. Do we understand this? So this is, each one is nucleosome, this whole is chromatin, condenses further to form chromatin fiber, and further condenses to form a compact structure called chromosome. Please tell me these points are clear. Now all these terms are used only for eukaryotes, and this condensation occurs only during cell division. Otherwise, you know that before cell division, that is in the interphase, this whole, it is in the chromatin stage. They don't condense until they are ready for division. Is that clear to all? Now guys, moving on to the structure of chromosome. So this chromosome has one chromatid, P arm and Q arm, we know that. And this is a primary construction called centromere, and around the centromere we have two disc-shaped structures called kinetochores. These are the site for attachment of spindle fibers, we have already discussed, right? And this is how DNA is super condensed within it, right? So there are histone proteins to stabilize DNA. But when we are forming the chromosome, so it is an NCERT line. So while we are forming chromosome, guys, we need some non-histone proteins also. So during chromosome formation, we need non-histone proteins as well. Is that clear? Now, during the condensation, please note that during condensation, there can be two types of condensation. Either some part of chromosome or some part of DNA, or chromatin will be loosely condensed, while the other part is very tightly condensed. Now the one which is tightly condensed is known as heterochromatin, and loosely condensed is known as euchromatin. Is that clear? Now what is the difference between hetero and euchromatin? U is loose. So because it is a loose thread, so you can detangle it. Think of it like, understand it like this. It is loosely condensed, so it can detangle, and against it mRNA can be formed. So transcriptionally active, and it is lightly stained. While heterochromatin is so tightly condensed that it becomes inactive for transcription, mRNA not formed, so not expressed, so protein synthesis does not occur for heterochromatin part, and it is darkly stained. Is that clear? Now remember, females have two X chromosomes. Out of these two, guys, one remains euchromatin and the other becomes inactive because it gets heterochromatized, and this is seen as a bar body. Is that clear? Now In case of prokaryotes, I told you, the DNA, see, DNA is moving around, sorry, DNA, yes, DNA is wound around these hook-like basic proteins. These hook-like basic proteins are called polyamines. These stabilize the bacterial DNA and they are in the form of hooks. Is that clear? And this is the bacterial DNA, which is double-stranded but circular, prochromosome or genophore. This is the difference between prokaryotes and eukaryotes. Please tell me that these points are clear to all. Is that clear? Now guys, what is Griffith's? Now we are going to search for genetic material. Let's again go back in the background of the movie where we don't know which is the genetic material, but something is there which is passing on the character from one generation to another generation. So brain is tracking, right? So now there is search for genetic material, and here we have three very important experiments. So the first experiment is discovering the genetic material, and that is called transforming principle. Earlier there was we did not coin the term genetic material. It was simply known as transforming principle. So it was this first step was done by Griffith, and see, the organism used streptococcus, or you can also use diplococcus. Then the chemical nature was not identified by them. Chemical nature of that transforming principle was identified by Oswald, MacLeod, McCarthy, that it is the DNA, right? They used the same experimental setup as used by Griffith, and organism was also streptococcus. They used streptococcus, but in some questions it is given diplococcus, so don't get confused. We will go with the diplococcus also. But even though Oswald, MacLeod and McCarthy in 1933 to 44, they established that, okay, it is DNA which is the transforming principle, people had their doubts. So it was still not confirmed. So there were some doubts, there were some questions raised. But the unequivocal proof, that means after which there was no tinge of doubt, that experimental proof was given by Hershey and Chase in 1952. You have to learn the name of the scientists, organism used, the year and their contribution. Is that clear? So this slide summarizes everything. Now, then guys, let's talk about the Griffith's experiment. So as said, what did Griffith do? Griffith's experiment, he took a mice, and mice were treated with different strains of Streptococcus pneumoniae. First, let's understand. The bacteria used is Streptococcus pneumoniae. We have two strains, R strain and S strain. R for rough and S for smooth. So what does rough and smooth indicate? So rough means there is no glycocalyx present, and smooth means it has glycocalyx. Now it is like an invisibility cloak of Harry Potter. Okay, so those which have glycocalyx, they will be invisible to our immune system. So basically, when the immune system cannot detect their presence in our body, what will happen? They will keep on hiding, they will keep on perpetuating, and they will keep on increasing their population and causing disease. So S strain is always virulent, while R strain, because it is easily observed by the macrophages, the immune system, because of no invisibility cloak. So what will happen? They will not be able to survive because WBCs will engulf them. So no disease. So remember, R strain is the non-virulent one and S strain is the virulent one. Is that clear? Now, since we have understood two different strains of the bacteria, what did Griffith do? First of all, he is going to infect the mice with the R strain. Now we know R strain does not cause disease. So our mice lives happily ever after. Nothing happens. Then Griffith was not happy. Griffith infected mice with S strain. S strain causes disease. It is virulent. The mice dies. Now he is happy. Who is happy? Griffith is happy. Now, no. Then mice was infected with heat-killed S strain. What did Griffith do in the third case? He took S strain, which causes disease, but he kills that S strain by heat, by heating it. Now what happens? When the S strain is killed, the mice lives. Yes? Nothing very different. It was all very expected, right? So all these things are expected. So Griffith is not happy with the expected results. he does this last experiment. Now he's going to infect the mice with heat-killed S strain kept with R strain. Remember. So what did he do? He took heat-killed S strain along with the R strain and he put this inoculum in the mice. What will happen? What should happen? See, if we go with the theory, R strain should not kill the mice. S strain, if it is heat killed, should also not kill the mice. So R strain and heat killed S strain, in both the cases, your mice should live, right? But what happens? When these two are combined, the mice dies. Are you understanding? The mice dies. And when the dead mice, when we tried to extract the bacteria from the dead mice, it was not heat killed, it was not R. The only bacteria which was extracted from the dead mice were all S strain bacteria. Now that takes his attention. Now Griffith is going to conclude something. So he said, I added heat killed S strain and R strain. But after the death of mice, why is it only S strain which is extracted? The living S strain which is extracted from the body. Is that clear? So he explained that why in this case the mice dies, that heating leads to opening of the bacterial pores. Something, he said, something from the S strain enters the R strain and transforms the non-virulent R strain into the virulent S strain which kills it. This is what he concluded. So he called that something. He doesn't know that what is that something, whether it is protein or fat or carbohydrate or DNA or RNA. He has no idea yet. He says that something is the transforming principle which transforms one form to the another. Like in this case, it transforms R strain into the S strain. So that is why he was only able to extract the S strain bacteria from the dead mice in this case. Please tell me that these points are clear. Yes, guys? Please tell me that these points are clear to all. Now, in the next case, guys, now Griffith laid the foundation. He showed us the path that, okay, there is something which can transform. But what is the nature? That was not identified then. This was done by 3 scientists, Oswald, McLeod, McCarthy, in the year 1933 to 44. It will take them 11 years. And they used the same Griffith's experiment or the setup. So what did they do? They took that Streptococcus bacteria, the S strain, right? So they took which S strain, and so they've taken these S strain bacteria, see, heat killed S strain with live R strain, right? And they've added some enzymes. So we know that proteases are going to dissolve the proteins, lipases for fats, carbohydrates for carbohydrates, DNases for DNA, and RNases for RNA. Now what did he do? After adding these enzymes, when he put this inoculum into the mice, so in this situation also mice dies, in lipase also mice dies. So basically fat is destroyed, mice dies, proteins are destroyed, still mice dies. So here carbohydrates are destroyed or digested, mice dies. So when DNA is digested, mice lives. When RNA is digested, then mice again dies. So who is the culprit? Who is responsible for transformation? It is DNA. So that's what these scientists explained. But the unequivocal experimental verification or proof that DNA is the genetic material was given by Hershey and Chase in the year 1952. So what did they do? The experimental organism that they used was bacteriophage and this E. coli. Is this clear? In one of the bacteriophages, guys, it was the sulfur which was radio labeled, S35 was used, and in other bacteriophage, they radio labeled the phosphorus, P32. Now we know phosphorus is a part of genetic material, you know, nitrogenous bases, phosphoric acid, right? So when we radio label P32, it is going to enter into the DNA. While if we radio label S35, sulfur is a part of protein and proteins are present in capsid. So this is going to radio label the capsid. Now when we infect this bacteriophage to the E. coli, when we infect E. coli with these radio-labeled bacteriophages, what will happen? So we know that virus is an obligate parasite. It is going to use the cellular machinery and multiply its number. So see, so new viral particles will be formed, right? New viral particles will be formed. After a while, we are going to blend this. So first step is infection. Second step is blending. We are going to blend it. And we are going to let it centrifuge. After blending, we are going to centrifuge it. What do we see? Whenever we centrifuge something, things settle down at on the basis of their density. The one which is floating is called the supernatant, and the other which settles down, that is called the pallet or the precipitate, right? Now see, in this situation, the supernatant contains the radio-labeled part. And in this case, it is a pallet that gets radio-labeled. So this proves that here the genetic material is not protein, it is the DNA. Is that clear? Now, properties of genetic material, see here. The properties are Rasme, so that is replication, stable, mutable and expressive. So the genetic material has to show these 4 properties. So any chemical which can be used as a genetic material has to show these 4 properties. So if we go by these 4 properties are only followed by RNA and DNA, not by proteins or carbohydrates or fats. Now see, what do we understand by replicative? Replicative means it should be able to form similar type of its own, right? Clones. So only DNA, only shown by DNA and RNA. So DNA can form DNA, RNA can form RNA, the process known as replication. Why? Because of the rule of base pairing and complementarity. What is rule of complementarity? A will always bind to T and G will always bind to C. This is rule of complementarity. Is that clear? This is the rule of complementarity. It should be stable. Stable means it should be able to remain as it is so that it can be passed on from one generation to another. Now, a very simple example of this is during the Griffith experiment, when he heat killed S bacteria and then kept it along with the R bacteria. And we know that heat killed S bacteria was able to transform the S bacteria. Why? Transform the R bacteria, sorry. Why? Because the DNA of the S heat killed S bacteria, the bacteria died, heat killed S bacteria, right? The bacteria died, but the DNA of during that heat killing did not destroy DNA. That heat killing did not destroy. It means DNA is stable. So it has to be that stable so that it can pass on from generation to generation. Another thing, 3 reasons which are responsible for the stability of DNA. See, double-stranded nature. So they are bonded to each other. So nothing else is welcomed to come and bond and react. So DNA is more stable than RNA because of double-stranded nature. Second, thymine is more stable than uracil, and sugar deoxyribose is more stable than ribose sugar. Please tell me these points are clear to you all. And then there has to be mutability. Means should be stable, but some variations should be possible, otherwise there will be no evolution. So both DNA, RNA are mutable. But as we said, DNA is more stable, hence it will be less mutable. RNA is less stable, hence it will be more mutable. Is that clear? And then it should be expressive. Expressive means it should be able to store the information and pass it on, and in such a way that some proteins are formed. So DNA forming mRNA, mRNA becoming proteins. So this is called the expressive nature. Now, if we go by all these things, we will notice that DNA is a better genetic material because it is stable. So it can store information from one generation to another. So DNA is more suitable for storing the information, while mRNA is more suitable for transmission of information. Are you getting that? mRNA is more stable. RNA of any type, less stable, more mutable. So it can only transfer the information. It cannot store the information over generations because, again, I hope these points are clear to you all. So what do you have to remember? Properties of genetic material. You should follow 4 rules: replicative, stable, mutable and expressive. Is that clear? Now, next point is central dogma. So flow of information. How does this information flow? So we know this was given by Francis Crick. The linear flow was explained by Francis Crick. What did Francis Crick say? DNA can form DNA, this is called replication. DNA can form mRNA, this is called transcription. And mRNA will then change into proteins, this is known as translation. So this linear flow is called central dogma or flow of information. So it is a linear flow. That flow, the information flows in one line, in one direction. This was given by Francis Crick. Watson had no contribution in this. But gradually, later on, when we studied some retroviruses, we discovered that it is not always the linear flow. There can be backward flow of information as well, that where mRNA can be changed into dRNA. This is called reverse transcription. It was studied in some retroviruses. This process is also known as termination after the name of Temin, who gave us this concept, reverse flow of information. Then talking about replication now. So what is replication? In replication, DNA to DNA is formed. Is that clear? The nature of replication is semi-conservative. Now what do we understand by semi-conservative? This semi-conservative nature was also established by both Watson and Crick. So semi-conservative means, so whenever a new DNA is formed, in that new DNA, one strand is parental and other is new. Every time, one of the parental strands will be conserved and the new strand will be formed in front of it. Is that clear? So this is semi-conservative nature. So let's say, see, this is parental strand. So they will separate, red will separate, and in front of that, one new strand will be formed. So this is semi-conservative nature. Had it been conservative, then the parental would have remained together and in front of that a new strand would have formed. So this would have been called conservative, but it is not conservative, it is semi-conservative. One is parental, one is new. Is that clear? Now this semi-conservative nature was established, it was mentioned by Watson and Crick, right? It is semi-conservative, but it was experimentally proven by Michelson and Stahel in prokaryotes. So in prokaryotes, it was Michelson and Stahel, and in eukaryotes, it was Taylor and et al. Et al means his members were there. So in eukaryotes, it was Taylor, and in prokaryotes, it was Michelson and Stahel. Is that clear? So the technique used here is cesium chloride density gradient centrifugation method. Organism used is E. coli, while Taylor is going to use faba beans, Vishia faba. Beans are plants, so hence eukaryotes, and they are going to use radioactivity method. Radioactivity, this was the technique used here. So can you compare? So be very careful whether they are asking for experimental verification of semi-conservative nature in prokaryotes or in eukaryotes. Is that clear? Now let's talk about the cesium chloride density gradient centrifugation method. So what happened in this method? This was done by Michelson and Stahel. So first step is that E. coli is grown in ammonium chloride. Now this is not a normal ammonium chloride. This is a special ammonium chloride where nitrogen used in ammonium chloride is N15, heavy isotope of nitrogen. We are using the heavy isotope. Now after growing E. coli in this heavy isotope containing, heavy isotope of nitrogen, ammonium chloride, now E. coli will be transferred to the medium containing normal ammonium chloride where there is no heavy isotope, it is N14 only. Is this point clear? Now, while we were growing E. coli in heavy isotope ammonium chloride, we centrifuged this E. coli. What did we observe? Centrifugation was done in cesium chloride, the solvent used for centrifugation. We observed that there is a bar, there is a precipitate, there is a palate at the almost near the bottom of the test tube. Is that clear? When we were growing it in medium containing this ammonium chloride N14, now there was this palate seen at an upper height of the test tube, almost in the middle. Is that clear? So gradually, now again bacteria continues to grow in ammonium chloride containing N14, and again when we centrifuge, we observe two palates, one at the same level as the previous one and the other at the top. Is that clear? So we are going to observe three different, see here, one near the base, one in the middle, and at the third level we observe two. Now see what happened. When we grew it in N15, right, we know the replication time of E. coli. So this is the replication time of E. coli, which is 20 minutes. After every 20 minutes, it is going to multiply. So here we had N15. So basically, both the strands of DNA will contain N15, N15. We call such DNA as heavy DNA, right? So when it was heavy DNA, we got a pallet to the near, towards the base of the test tube. Why? Because it is heavy. What is the name of the method? Density gradient centrifugation, right? So you are not checking the radioactivity here. You are checking the density. We know N15 is heavy isotope. So the more the heavy, it will be towards the base of the test tube. Heavy is towards the bottom, right? Now, when we put it in ammonium chloride containing N14, what will happen? We know it is semi-conservative, so these two will separate, right? And in front of that, one new parental strand will be formed, and now the new strand will contain N14. Do we see? So the DNA containing 115 and 114 will be known as hybrid DNA. So of course, it will not be as heavy as this. So intermediate, so you get a pallet in the center of the, almost in the center of the test tube. Is that clear? This is the hybrid DNA. Another 20 minutes pass. So in total 40 minutes have passed. What will happen? This will also segregate. So red goes separate, white gets separate, and white represents N14, right? 14 and N14 and 14. So do you see how these are formed? So this will be hybrid and these two will be light. Those containing N14 and 14 will be known as light DNA. This is known as light DNA. So the lighter the density, it will be present at the top of the test tube, and the solvent we are using is cesium chloride. That is why cesium chloride density gradient centrifugation. Is that clear? So now see here. So this confirms that it is a semi-conservative nature. Is that clear? Now here also we get to solve some questions. So we'll be solving those questions in the special classes. So I've already told you this in eukaryotes. Now talking about the mechanism of DNA replication. So we know it is semi-conservative. How DNA replication occurs? So for that, guys, the machinery used, number one, is ORI, origin of replication. So it is the site of DNA from where replication begins, and second, it decides the copy number. Second, we need DNA dependent RNA DNA polymerase. Now see, let me just okay. Now see here. So what does DNA dependent DNA polymerase does? DNA dependent DNA polymerase, it will look at DNA and form new DNA. DNA dependent. So this polymerase will depend on DNA, look at it, read it, and form new DNA according to that. Is that clear? Remember, this polymerase, DNA dependent, any polymerase, even later on when we will talk about RNA polymerase, it will always read in 3 to 5 direction, and and it will polymerize in 5' to 3' direction. Is that clear? Now, and we need a DNA template. So both the strands will act as template here. Are we clear? We'll need some other enzymes also. We'll talk about them gradually. Sorry. There is no. Okay, I'll add it here. Now see here. What happens? First of all, yes guys, so first of all, so this is the DNA strand, right? So this DNA strand, let's say this is 3'N and this is 5'N, this is 5'N and this is 3'N. So both the strands, they will act as template strands. This is also template, this is also a template. Now first of all, the whole DNA is not going to unwind in one go itself. So even before this, guys, the DNA was in the helical manner. So DNA is supposed to be in the helical manner. So this helical DNA will become this, unwind in the presence of enzyme helicases. It will become unwind in the presence of helicase, or we call it unwindase. Is that clear? And then it will start separating. So the hydrogen bonds between these, they will break. Now what happens here? The next step would be that this DNA, this will wind, it will unwind like this. So it has unwinded by the help of helicase. Now it will open up. Now the hydrogen bonds have broken. So the whole of it will not break or open up. So this is the formation of replication fork. This Y-shaped structure in which the replication is going to occur is known as replication fork. This is a Y-shaped structure. Now see guys, here. So let's say this is 3' end and this is the 5' end. Is that clear? Now, because the two strands are complementary to each other, what will happen? They will try to come back. So here we need some enzymes or proteins to keep them separated. So these are single-stranded binding proteins, so they prevent them from rewinding. Also, we will need some other enzymes, topoisomerases. If the DNA is highly coiled here, right, then topoisomerases are going to unwind it. So we need helicase or we need topoisomerase depending upon the coiling of the DNA. Now here we'll need SSBPs to keep them separated. Now look very carefully. What will happen? So the first thing, we'll need an RNA primer. So primer will bind here, or DNA polymerase will come in first. So DNA polymerase, we said, see here, what have we said? The DNA polymerase will always read in which direction? 3 to 5. Let's go back then. 3 to 5. So polymerase will always bind at which position? 3' end. So we know if this is 5, this is the 3' end, right? So DNA polymerase, this is the DNA polymerase, will bind on the 3' end, like here. And on this strand, it will bind on this side. So this is, and it will start reading the DNA from 3' end to 5' end. From 3' to 5' end. Please tell me that these points are clear. Right? And while it keeps reading, the new strand will be built in this direction. So the new strand will have polarity 5 to 3. That means new polynucleotides are polymerized in which direction? 5' direction to 3' direction. Is that clear? And in this case, it is going to go in this direction. Yes? Please tell me these points are clear. So both the strands will be known as template strands. But guys, now see, in this case, what will happen further? So now when it opens more, see, gradually it is going to open even more. It is going to open even more. So just see here. So we had already formed, in this case, we had already formed a new DNA strand, right? While it was open, this was formed. And this has already formed till here. Yes or no? Right? There was DNA polymerase and this was the DNA polymerase. This one has already formed in the previous step. Now what happens? The replication fork opens further. Now this strand was formed. It has opened again. So now in this situation, DNA polymerase will have to come back here, read it again from 3' to 5 and form another fragment here. Yes or no? Right? While this was already going in this direction, so when the replication fork opened further, what happened? Nothing. It just went on increasing in the same direction. So now do you see here that although both the strands will act as a template strand, but one will be known as a continuous strand or the leading strand, and the other one will be known as discontinuous or the lagging strand. Right. Please tell me that these points are clear to you. Please tell me these points are clear to you. Sorry. So this is 3-n, this is 5-n, this is 3-n, and this is 5-n. Please tell me these points are clear. So here these fragments are known as Okazaki fragments, and these Okazaki fragments, in the name of Japanese scientist, they are connected through. So there will come DNA ligase here. Now DNA ligase will come in, and DNA ligase will join the two Okazaki fragments. So these will be formed in fragments while this will be continuously formed. Please tell me that these points are clear. So this is how replication occurs. Are we clear? Are we clear? Yes, please. Quickly let me know, guys. Are we clear? So the polarity of the continuous strand is known as 3' to 5' guys, and this one will be known as 5' to 3' end. Please tell me that these points are clear to everyone. Yes, please. Is that clear? So replication will not start from anywhere random. It will start from a specific DNA sequence which is called ORI. So what do we need? We need ORI, we need this enzyme, we need topoisomerases for supercoiled DNA helices, we need SSBP, single strand binding proteins, so as to prevent the rewinding of the two separated DNA strands, and we need DNA ligase in case of for joining the Okazaki fragments. Are we clear till here? Yes. Now guys, moving on to transcription. So what is transcription? In transcription, we are going to form RNA from DNA. So it can be all types of RNA forming from DNA. Is that clear? So we have RNA to DNA. It could be mRNA, it could be tRNA, or it could be rRNA. Now, unlike in case of DNA replication where both the strands were acting as template, in case of transcription, guys, only one will be template and other will not be used, right? So now the template one is known as non-coding strand or antisense, and the other strand against which mRNA is not formed is called coding strand or sense strand. Now, how do we identify which one will act as a template or which one will be acting as a coding one? For that, we have to understand the transcription unit first. So see, the transcription unit is first formed of three units, three things. One is promoter, this is the first. Second is the structural gene against which actually the mRNA has to be formed, and the third component is terminator. Now, it is the promoter which determines. Now, these lines of NCERT are very, very important. Now, see here. So, it is, and this in 3rd May paper also, we got this whole paragraph as statement-based statements. Now, see, so it is the position of the promoter which determines which strand will act as template. That means non-coding, and which one will be coding. Is that clear? Now, before we understand how, please understand few terms. That 3' end is known as downstream, guys, and 5' end is known as upstream. Is that clear? So again, this is downstream, this is upstream. Is that clear? 5 is higher, upstream, 3 is lower number, so call it downstream. Now, who is going to determine which is the coding and the anti-code or the non-coding? It is the promoter. Clear? Just check the location of promoter. If this is the location of the promoter, now towards the promoter, the one which has 3' end, towards the promoter, the strand which has the 3' end will be known as the non-coding strand or the template strand. Is this point clear? So who determines? It is the promoter. How do you determine? The strand which has 3' end towards the promoter side is the non-coding strand. Yes? Can I say promoter is present towards the 3' end? Sorry. Now, non-coding is decided. Against this non-coding, mRNA will be formed. Now, what is the significance of coding strand? So, when you have to tell the location, you know, whenever we have to tell the location of some place, we always use a landmark. So, now, coding or sense strand is used as a reference for location. Now, can I say, what is the location of promoter? Promoter is present towards the upstream or 5' end of coding strand. Do you get that? I'm repeating, guys. How do you identify which strand is the coding and which one is the non-coding strand? See, for that, first of all, you have to see the location of the promoter. So, promoter determines which one will be coding and which one will be non-coding. Step 1. So, identify the promoter. Second, look out for the 3' end. So, the 3' end towards the promoter side. Now, this strand will act as a coding strand. Do we understand this? Yes. Now, what is the significance of this one will act as a non-coding strand. Yes or no? Please tell me these points, guys. I'm repeating once again. This is very, very important, guys. See here. So, if you have to identify which one is coding, which one is non-coding. So, first step is you identify the location of promoter in the transcription unit. Is that clear? So, once the location of promoter is identified, now for example here promoter is in this direction. Now here look out for the 3' end. The strand which has 3' end towards the promoter side, now this strand will act as a template strand or the non-coding one. Is that clear? What is the significance of coding strand then? So, significance of coding strand is it will be used as a reference for the locations. Yes? Can I say, if somebody says, what is the location of promoter? Can I say promoter is towards the 5' end of coding strand? Or similarly, what is the location of terminator? Terminator is towards, so this is terminator. So, terminator is towards the 3' end of the coding strand. Do we understand this? Now, third point, instead of 3' end and 5' end, they can use the terms upstream and downstream. So, 5' end is called the upstream, 3' end is called the downstream. Are these 3 steps clear? So, that is how we identify which DNA strand will act as a template or the non-coding and which one will act as a coding or the non-template strand. Is that clear? Now, what are the requirements for see, these are the transcription unit 1, 2, 3. Promoter, structural gene, terminator. Told you promoter is towards the 5' end of the coding strand, terminator is towards the 3' end of the coding strand. Yes, and in between region along which or opposite to which mRNA will be formed is called the structural gene. So these are the transcription unit. What are the other requirements for transcription? For transcription, we need enzyme RNA polymerase, DNA dependent RNA polymerase, and we also need some additional regulatory proteins like sigma factor and like rho factor. Is that clear? Is that clear to all? So how does transcription occur? See, it will begin from promoter, that is why we call it promoter, and it will end at terminator. So RNA polymerase will be required in all the 3 steps. First step is initiation, then we have elongation, and then the end, that is termination. These are the 3 steps. What are the 3 steps? Initiation, we have elongation, and the third step will be end to it, that is termination. In all the 3 steps, guys, RNA polymerase will be used. But now RNA polymerase will require some additional regulatory proteins to know whether to start the process or stop the process. Now, in order to initiate the process, guys, RNA polymerase will bind with sigma factor. S for sigma, S for start. It will start with the sigma factor. Then after initiation, sigma factor will separate and only RNA polymerase is enough for elongation. Is that clear? And then termination, RNA polymerase will bind with rho factor. R for rho, R for rokna. Rokna means stop. So R for rho factor. Rho's factor is going to terminate the process. This is transcription. Is that clear? Now, see, you get certain questions where you have to write down the complementary strands. So how do we do that? So first of all, if such structure is given where they have identified the promoter and the terminator, so first of all, you will have to find out which one will be the template and the non-template strand, or non-coding and coding strand. Now, for example, promoter determines this. This is the promoter. So towards promoter, the 3' end, the strand with 3' end, this is the non-coding. Yes, this is the non-coding, and one opposite to it is the coding. Now, can you write down the complementary base pairs here? Can you do that? Yes. So just see the conclusions. So template strand is 3 to 5' end. The complementary strand will be, mRNA will be complementary and will have opposite polarity, while the coding strand will have same reading and same polarity to that of mRNA. Just that T will be replaced with U. So you will use these conclusions to write down the answers. And I've told you the 3 steps: initiation, elongation and termination already. Now, what is the difference between transcription in prokaryotes and in eukaryotes? See here. So as in prokaryotes, for transcription, only one type of RNA polymerase is used to form all mRNA, tRNA or rRNA. And we have polycistronic genes. That means one gene will form many proteins. So transcription occurs in the cytoplasm because there is no membrane-bound organelles, no nucleus present, right? And then in prokaryotes, the whole DNA, there is small amount of DNA present and the whole DNA is expressive. There are no non-expressive regions present. So there are no introns. Is that clear? Therefore, we don't need any post-transcriptional changes. Is that clear? Now, the mRNA formed in front of DNA mRNA formed, this will be fully functional. It will be of mature type. We don't need any changes. So as this mature mRNA is formed, many ribosomes will come and attach itself to the mRNA chain. So polyribosomes will be formed here, and because it is already full and functional mRNA formed, so protein synthesis will occur simultaneously. Okay, so we say that transcription and translation are coupled together. So here single mRNA has many ribosomes, which forms a polysome. Why? To form multiple copies of the proteins. Why? Because we have very small amount of DNA here. So it increases the efficiency. Now, if we talk about transcription in eukaryotes, so RNA polymerases are of 3 types, and different RNA polymerases are going to form different types of RNAs. We'll study which ones. It has monocistronic genes, means one gene will form one protein only. Now, the location for transcription in eukaryotes is within nucleus. So you see, first of all, the DNA in eukaryotes, it has a split gene arrangement. So the entire DNA in our body, in our cell, is not useful. It is not functional, right? Not expressive. So the expressive one, that means against which mRNA will be formed and protein synthesis will be proteins will be formed, that is called exons, and the non-expressive is called intron. So E for exon, E for expressive, remember that. So we have a split gene arrangement. So say exon interrupted with introns and so on. So first of all, the mRNA which is formed, it is formed against the entire length of DNA, that is both for introns and exons. Yes. So this mRNA formed, which includes regions for exons as well as introns, this is called heterogeneous RNA. It is not mature, it is not functional. So it needs some changes. Is that clear? It will need post-transcriptional changes. We'll have to remove the mRNA formed in front of these intron regions. Is that clear? So we'll have to splice it, we'll have to cut it. So this step is known as splicing. Also, we'll have to put a cap here on the 5' end and put a tail on the 3' end to secure that mRNA. Why? Because mRNA formation and protein synthesis is going to occur at different times, and there is a time lapse between that, during which mRNA can get degenerated. So in order to protect that, we have to cap it, we have to tail it, and we'll have to cut the unrequired or the non-expressive regions. Is this point clear? So the post-transcriptional changes, one is splicing, second is capping, third is tailing. What is splicing? Removal of introns. What is capping? This is addition of, capping is addition of 5-methylguanosine, and it occurs on the 5' end. Tailing, T for 3, T for 3' end. It occurs on the 3' end. And what happens? We add polyadenylate residues to it. So guys, these are the differences you can go through later. And now, very important, there are 3 types of RNA polymerases. For RNA polymerase 1 is going to form rRNA, and the different types you can see here. RNA polymerase 2 forms 2 types. One is hnRNA and second is mRNA. This is heterogeneous, you can call it precursor or nascent, and it is not functional. And 3, T for 3, and it is going to form tRNA, and 3 types. tRNA, 5S rRNA, and snRNA. These are the 3 types of RNA polymerases used for the formation in case of eukaryotes. Now, moving on to post-transcriptional changes here. See, in post-transcriptional changes, guys, we are doing 3 things. I've told you. Capping on the 5' end, where we add 5-methylguanosine. Splicing, in which spliceosome will be formed. So the intron region is going to come out, evaginate, and form a spliceosome, and then it will be spliced like this, right? So that is why the real or final mRNA is much smaller than the heterogeneous or the DNA transcribing. And tailing is done on the 3' end, where we add 200 to 300 residues of polyadenylate residues. Now these changes are needed only in case of eukaryotes. Do we see a cap, a tail, and splicing of introns here? Now guys, now we come on to very important aspect, that is genetic code. See the nature of genetic code, triplet nature. Every code has 3 bases, AUG, CCC, UUU, whatever. Then unambiguous means one codon will always code for the same amino acid. Then it is non-overlapping. So one base is only a part of one codon. Is that clear? So if we have this long chain A, U, G, C, C, G, 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 A, and U, let's say. So it is always, each base is only a part of one codon. So this C is not a part of another. There is no overlapping. Is that clear? So it is non-overlapping, almost universal. So if UUU codes for phenylalanine, it will code for phenylalanine in almost all the organisms. Is that clear? Comma less, punctuation less, or there is no punctuation here. It is continuous, right? We don't put commas or bars in front. Then degeneracy, very important. One amino acid can be coded by multiple codons. Is this clear? Now, genetic code, main contribution was by Marshall, Nirenberg, and Herb Gobind Khurana. Now see here. So 3 specific nucleotide sequence that codes for a specific amino acid. This 3 specific nucleotide is known as a codon. Is that clear? So you have to remember. Now see, every protein synthesis or translation will start from AUG. So AUG is known as the start codon. Is that clear? While it will end at these codons. Whenever you find UAA, UGA, UAG, these are the stop codons. These are 3. Is that clear? These are some. So it means we are going to have anti-codons for only 61 codons, because so 61 are codons which code for amino acids, 3 are the stop codons because they don't code for any amino acid. While AUG has a dual function, guys, it is a start codon as well as it codes for amino acid called methionine. Are we clear? Please tell me. So the triplet nature, guys, was determined by a physicist named George Gamow. Is that clear to all? Is that clear? Right? So this is the genetic code. You don't really have to learn. And now, guys, see, let's talk about the translation unit. Now, talking about the translation unit. So we know that translation unit consists of three things. This is the start codon, and then UU. So there are three stop codons. So it starts with the start codon. We have a stop codon and we have the intermediate part, right? So these three things are going to form the translation unit. Is that clear? Now, there is a part of mRNA before start codon. There is a part beyond this. Now, this region is called UTR, untranslated region, present at 5' end and 3' end before start codon and stop codon. So, UTR flanks on both the sides. Why? It protects, it increases the efficiency. It protects the part of mRNA which actually has to code for proteins. Is that clear? So see this. So what are the requirements? We need RNAs, we need ATP, we need peptidyl transferase, start codon and stop codon. Is that clear? So guys, it is already time. Let's stop this here. Tomorrow we are going to talk about biotechnology, and before biotechnology I'll discuss protein synthesis as well with you. Is this clear? So tomorrow also I'll meet you at 7. Today the class got delayed because of some work going on here. So I had to rectify this light wavering which happened in the previous class. So we'll start the class tomorrow at 7 p.m. and we'll be able to complete some remaining part of this chapter, DNA fingerprinting, very important, and biotechnology. Is that clear? So thank you so much. But don't forget, 7th July, you have to take the AIMT, and I'll see you tomorrow at 7 p.m. Bye, guys.